This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 638, recorded on July 10th, 2020. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from New York State, Daniel Griffin.
Hello, everyone.
And also from New York State, Chuck Knirsch.
Hi, Vincent. Hi, Daniel.
Here we are. A week goes by very quickly, doesn't it?
It does, actually. I feel like I start preparing for this TWiV talk, a few minutes after we finish the last one. Yeah.
I meant to tell you, I really appreciate that you obviously prepare, and you've thought about what you're going to say, and it shows. People like it. So thank you for that very much.
Oh, certainly.
Okay, we are in the first week of July. What's going on, Daniel?
Yeah, so I'm gonna start with a quotation again, and this one's sort of timely. And so this is a Vincent van Gogh quotation. And thanks to the pandemic, I did not get my family trip to Amsterdam. My family are all Vincent van Gogh fans, and we had this planning for a long time, and yeah, that didn't happen. And so here it is. The fishermen know that the sea is dangerous and the storm terrible, but they have never found these dangers sufficient reason for remaining ashore. And I say that in part because, I'm going to go race sailboats after this. So that's one good thing. But the other is, there's a pulmonologist I've worked with for a long time, a really entertaining, very passionate man. And actually the other day he was saying, if these numbers go up the way people are predicting this winter, I may just retire. I'm not that young anymore. And I think he was overwhelmed in a lot of ways during the first, period of months. But I guess this is my sort of encouragement to all the other healthcare providers out there, stick in there. We really need you. I mean, I know this is tough, and a lot of us are anticipating it's going to be really tough again. And in a lot of areas, it's really tough right now. There's some areas, Florida, Texas, other areas where the ICUs are filling up. And so just sort of, remember at some point, this is why you went into medicine. You went into medicine to make a difference. And I know it's tough, but this is when we can make that difference. Doctors, nurses, all the people that are involved. So that's why I use that quote there. But next, I'm going to say thank you again to everyone who's been helping us support FMRIC in Uganda. That has gone really well. So visit parasiteswithoutborders.com and keep helping us. We're only about $7,000 away from our goal of being able to give them $40,000. So please jump there. But let me go through what I like to do is as the week goes on, people ask me questions. And then I figure if clinicians are asking me these questions, they're being asked these questions. And I try to give a response to what might be causing this. So one of the questions that actually was coming up quite a bit this week was, why are the case numbers rising, but why aren't the deaths rising? Right. And I know a lot of people have been asked about this, but in the media, I heard that Anthony Fauci is going to be on TWiV next week. So I know he had some answers to this. And I'm always curious, are my answers and Tony Fauci's answers the same, right? So if they are, that's a good sign. And I think a lot of this, I think we're on the same page. One thing that he points out that I will echo is that we are seeing younger people infected at this point. Early on in New York, the mean age was in the 60s. We're now seeing that mean age drop down to the low 40s, right? And, we've said all along, one of the best things you can be if you're going to get exposed to COVID is a young woman, but young is key as part of that. So when younger people get infected, the death rate tends to be lower. The other, and I think this is true, and this is something, is that we are better at managing patients. Even if you look at people the same age that are ending up in the hospitals, they're much less likely to die. And I think a lot of that is interesting. We're starting to get the randomized control trials, but a lot of things we sort of stumbled onto, we arrived at initially observationally. So, delaying the intubation, the proning, steroids, right time, right dose, right patient, anticoagulation, which I really think made a made a big difference, continues to make a big difference. It was just another paper out showing that the exact number was 37% of the patients they looked at had pulmonary emboli. So this was huge and devastating. And then I think we're also avoiding that kitchen sink of toxic cocktails, right? I said early on, like, if I can't have a cocktail at work, I don't want my patients to have cocktails either. I think it doesn't help with my job performance or their outcomes. The other, and I think this is big, and this is a bit controversial, and I'm sort of going to step into it, I think here, is there's been a lot of investigation, looking at New York, and is it a witch hunt? Is it, let's learn from what went wrong so we don't make the same mistakes? But a lot of the deaths, as I think people know now in the New York area, were in the nursing homes. Actually, about half of the deaths, right? And how did that happen? And so we had a pause order that went into effect March 22nd. Would have been great if it had happened sooner, but that was the whole what went wrong and the testing delays. But once we had identified community spread in New York, and saw how many thousands of people were infected, that went into effect. But just a few days later, March 25th, the governor was actually advised, recommended. He made an executive order and said, listen, our hospitals are going to get overwhelmed, so nursing homes have to take these COVID infected, potentially contagious people back. And the actual wording, right, was no resident shall be denied readmission or admission to the nursing home solely based on a confirmed or suspected diagnosis of COVID-19, actually prohibited requiring hospitalized residents from getting COVID-19 to affect the admission or readmission. So that finally on, I think it was May 10th, actually, was removed. That executive order was rescinded and we stopped doing that. But that was a disaster, right? Looking back on it. And I think, you look at Florida and you say that is a place with thousands of nursing homes. Why aren't the nursing home residents dying there? And they've actually not done this. They've been really good about testing staff, of protecting vulnerable populations. So, I know right now there's this big, who gave the governor that advice. But the big thing is that advice we now realize didn't allow us to protect a vulnerable population, and that had a big effect.
We also did the testing in New York at the time that we do have in Florida now. So maybe a little bit of difference in time period as well as a policy, error, I think.
Yeah, I think that's right. And I think that what I was worried about, which hasn't come to pass, is there was actually pressure, right, back to the governor saying, even though we can test now, let's not wait for negative PCRs before we send them back, sort of a pressure to just pick a random date and say, oh, maybe 10 days, maybe it's 21 days. We're just going to say, oh, they're not infectious and send them back. And fortunately, fortunately, the governor has held firm until I think we're sure that people are not infectious. We don't want to send them into a setting. I had a question recently in one of my roles in the sports world. If you have an athlete who's PCR positive, are you going to send them back to training, back to be with the team? And, for me, I was like, this is an easy answer. I know that it's horrible for that one player who's still PCR positive. But if you send that person back to, we'll use the NFL as an example, back to the Giants, and now all the Giants are sick and you shut down the NFL, that's a disaster. Well, I'm going to say it's the same if you send that person to a nursing home and all the people there start getting infected. And then the fourth, and I think this is true, is that, it takes time to die. Right. So, we will probably start seeing an uptick in deaths in the next week. So I anticipate, a week from now when we have our talk again, the rates of new cases already broke 60,000 yesterday. I think the deaths will be above 1,000 just because that's sort of part of the dynamic. Yeah.
Have your admissions gone up this week?
Remember when I recorded last week, I had nobody in the hospital. Yeah. Now I had a couple people in this week, and one of them I'm going to talk about because he's a very interesting character. Testing. We're having challenges again, big challenges. One is we're having issues with getting the proper supply. So I was talking to, actually the chief of our pediatric division at, ProHealth New York, Jay Berger. And he was saying, no, it was definitely a good sample because the man said they shoved that Q-tip all the way back and it was incredibly painful. I'm like, what kind of Q-tip did they shove back? And, we were having issues getting the proper Q-tips, the small ones where it feels like a tickle. So they were taking an oral Q-tip and shoving it back to the middle turbinate. And that's just not, that's cruel and unusual. So, and then we've had issues, I think I talked about last time, is delays, resulting delays. And I'll say this publicly, it can be an up to an 8-day turnaround in New York at some places from the time you procure the test to the time you get a result. And that's a disaster from identifying infectious days and stopping the spread.
The commercial labs are reporting that they've had a doubling in their turnaround time just because of the volume, I think. So,
yeah. Yes, I mean that continues. We're everyone can have a test eventually as long as they don't mind getting the results by Christmas, right? So this is frustrating. The other I want to talk. This is something that keeps coming up. The concept: does natural infection with coronaviruses cause herd immunity? Does it protect us from reinfection? And so we got an email last time from a physician down in Washington, D.C. So I followed up on that. And I'm also going to discuss an individual that I just took care of. I'm taking care of currently. So the individual that I just, readmitted this weekend is a gentleman who I took care of in the end of March. He's actually a friend of mine, a fellow clinician. And he contacted me in the end of March. He was sick. His family was sick. We arranged for everyone to get tested, right? That was a Herculean task at the time. But they were all confirmed PCR positive. I took care of him in the outpatient setting. He was sick for a couple weeks and finally, was better, returned to work. And he looked like he'd been through quite a bit at that point. But then he was better. He was out doing his job, seeing patients. Last Saturday, fevers, chills, wasn't feeling well. We ended up readmitting him. And we did a COVID PCR. It's 4 months later, and that COVID PCR was positive. This gentleman, interesting, I'll add this in, he had such high antibodies back in April that he was actually a plasma donor for Mount Sinai. Repeat serology testing, and his antibodies are barely detectable. And he had his neutrophil lymphocyte ratio was in the 30s. His D-dimer was 1300. His interleukin 6 was up in the 30s. He actually also has bacteremia. He's got what looks like an infected finger. So, how much of this is the bacteremia, but I'm not sure bacteremia gives you a positive COVID PCR 4 months out. But I called up and I spoke to the person who emailed me, just sort of, because one anecdote is an anecdote, two anecdotes are two anecdotes, not data. But I spoke to the gentleman about the Washington, D.C. case, and it was very similar. It was a gentleman early April, got sick, PCR positive. Middle June, his son was sick, and he thought, as I think a lot of people, I've already had this, I should have protection. A week later, felt sick. 2 weeks later, sick enough to go to the ER. COVID PCR positive after having a couple negatives to go back to work, and sicker the second time than the first, right? Just like my individual. First time outpatient, not a problem. Now in the hospital. So starting to worry about the idea that this, like every other coronavirus, may not give long-lasting immunity. So herd immunity from natural infection may be something beyond us. And as I talked to my colleagues, I talked to a different pulmonologist today, and he was saying, I've seen a bunch of people and they seem sicker, and I would have thought it was the COVID, but you really can't get that twice, so I haven't been testing them. So I've noticed that a lot of doctors, instead of saying, I don't know, have been reassuring people who have positive serologies, saying, well, it looks like you've got some protection here. I think, we have to be honest and say, we don't know. And all strategies where we're saying, oh, well, if people get infected, they go to parties. If it spreads the next time around, well, at least a portion of the population will be immune. From the studies with antibodies dropping from a couple of these experiences that we're starting to have, I'm starting to get a little bit worried, so I'll put it there. Vincent, get that hand up.
So, these are two cases, but maybe there are, for these two, there are 200 where people recovered and are not getting very sick. So you're going to see that, right? Always in infectious disease where a lot of people are infected. So maybe it's not as bad as you're thinking.
I hope not. I hope. I mean, the tough thing, right, is that if you look at the common coronaviruses, they don't give us long-lasting immunity. Yeah. And they traditionally, you could be reinfected, multiple times in the same year. There's a preprint interesting coming back out because you have to publish something after 10 years, it becomes invalid. But, we've known this for the other coronaviruses that if you get, OC43 or 229E or any of the other, you don't get lifelong immunity. You can get sick again. And it's not clear that that second infection is milder than the first. So just my word of caution to be careful about, when we hear that 5 or 10% of the population is seropositive, I don't know what that means yet.
What about recrudescence, though? And I guess we need the virus before the first virus and the second virus, right? Because it wouldn't be unheard of to have a viral illness have it with recrudescence as well.
Yeah, that's interesting, right? When I looked at the literature again, I was like, how do we even know what the old, when we saw the same, like we do these rapid viral panels, when we said, oh, you had this 2 months ago, it's coming up positive again. How do we know that isn't just that coronaviruses have this recrudescence? Yeah, I don't know. Well,
we're learning in it. Sorry, Vincent, go ahead.
If you had virus isolates, you could sequence them because that would tell you they should be distinct, right?
Yeah,
Right.
and I would love that. So, sort of a call out to our listeners. Someone please let us know because this is really has a big effect on how we approach this, right? I was asked a month or so back. We were designing large treatment trials, vaccine trials, and trying to pick where to do it, right? So you want to predict a place where there's going to be a large amount of infections. And I actually was suggesting Chicago because in my head I was thinking because I don't know, it may be that New York. That there is some of this herd immunity and we don't get it as bad the next time. But again, I don't know. This will be something we find out. I know I've had a lot of people say that it would be great in addition to the serology to have a T-cell assay. So we would love to have that, by the way. Researchers who are listening, the clinicians would love to be involved in studies, looking into that as well. And I guess I'm going to close on this last one. I get a lot of questions about the school reopenings. And this is a tough one. Vince and I were chatting just briefly before we started here. And it's hard. I mean, I think there's a lot of us, even the American Academy of Pediatrics, that are saying the goal should be to open the schools. But I don't think any of us are saying the goal is to open the schools in a way that is unsafe. And I was using the analogy. They're saying, oh, it's safe to drive, but we've taken away your brakes and your seatbelts. It's not safe to drive without brakes and seatbelts. So if we're going to get kids back to school, it's got to be done safely. It's going to require testing. And we have a number of papers out looking at what that frequency is going to be. And the reason I say it's going to require testing is, we're learning, and sometimes it takes us a while to learn, but even people who become symptomatic, probably half of the spreading events occur before the symptom onset, certainly before they know and get a positive test. And then people who are asymptomatic and are spreading while asymptomatic. So without testing, without surveillance, this is not going to go well.
You mentioned the average age going down to the 40s, right?
Yes.
Is that the average age of infection or serious disease?
It's the average age of people who are diagnosed. I think we've seen pretty consistently that if you look at people who end up getting admitted to the hospital, right, it's older people. I think we talked last time about you look at people who end up in the outpatient setting. Well, they're younger. It's sort of the reverse of that.
Okay.
We're seeing consistency as far as the people who end up admitted tend to be the older individuals.
Okay. All right. I wonder if I could have both of your opinion on this brain paper that came out this week describing a range of neurological findings like encephalomyelitis and neurological disorders. Guillain-Barré. What do you guys think? Are you seeing this, Daniel?
I think I tried to share a bit of this early on. We certainly in a number of patients saw the Guillain-Barré. I think I may have asked Dixon to help me pronounce that early on. But we would see, and it made sense as far as the timing would go. This would be about week 3 when we would expect sort of a robust antibody response. We think of this disorder as being antibody mediated. But there also, we saw delusions. There was a nurse, actually. She was an ICU nurse who ended up in the hospital. And when I first saw her, she was, anytime you would touch her, she basically would start screaming, I'm falling. And it was really quite frightening. And then the next day she was like, oh my gosh, I feel so bad about how I acted. I was like, no, it's fine. But she was, and we saw this throughout. We saw it in ICU patients, right, who would end up extubated, and there was this fog that, would last for a while. And then even patients that, I think I described in the long haulers, people describing it or saying, I have this brain fog. It's just not as sharp as I used to be. It takes me a little time. So strokes, we certainly saw. That was one of the, sort of red flags that even outpatients are susceptible to thromboembolic phenomenon. So I think they described the delirium, the psychosis, which we certainly saw. What we thought was, an inflammatory process. I think we saw that. The Guillain-Barré. So yeah, I think everything they're describing here was consistent with our early experience.
All right. So this is just a quantification in their area of their patients. And I think, as you mentioned earlier, none of this is related to virus being in the brain. It's probably secondary to immune responses, right?
I think so. I mean, if you think Guillain-Barré, we've always sort of thought of that as an immune response, and they respond quite well to intravenous immunoglobulin, so IVIG, steroids. The strokes, again, we think is a thromboembolic. So yeah, so I think these are immune thromboembolic, other complications, not the virus. At least that's my impression to date.
Story in the Times today, 68% of patients in a storefront medical office in Queens were seropositive. Now, based on your discussion just now, I'm not sure what that means, but that's pretty high. And you think it just happens to be that these are people on the front line that are working and they're getting infected?
It was interesting because it was actually an ER doc who wanted to talk to me about this just today. Yeah. He's an ER doc who got sick in early March, but never seroconverted, stayed antibody negative. And so he brought this up with the idea, look at these people in these communities, they're protected, they're going to be fine. They won't have to worry about the next wave. And I was like, yeah, I don't know about that. And so that's interesting because people take that. And in certain communities, I think there was a large percentage of people that ended up getting exposed. The big issue for us, right, is what does that mean this coming winter?
I hope they're protected because, the borough of Queens was just devastated, and Elmhurst Hospital in that area was just overwhelmed. So I'm hoping they have some community protection for this winter.
I mean, I just don't know any virus that infects you and then your immunity is gone in 3 months and you get infected in serious disease. Do you know of any such virus?
Well, I was thinking about several different pathogens. I'll say we'll start with viruses, right? Like you don't get herd immunity to dengue, right? Like every year, right? There are 500 million. If anything, if your antibodies drop to the wrong level, people don't do well. So malaria is a classic, right? You get exposed. If you stay exposed, you do okay. But then boom, some, HIV is probably perfect, right? You don't get exposed to HIV and become immune. Yeah. So I think there's a number of pathogens that are like this. And I think that's, as a viral strategy, right? This is a very consistent genetic code. So, influenza, which transmits in a similar pattern, a lot of pre-symptomatic tread, it can change its genetic code. So it doesn't really care that you get, antibodies to that strain because then the next strain. But this one, for these coronaviruses to circulate, they need to basically somehow succeed in not creating long-lasting immunity, or they die out, they become a low prevalent infection. So Chuck, I don't know if you had any.
We need to study it. I don't know. I'm hoping that this one is more like SARS-1, where we do seem to have more persistence of antibody. We didn't have the infection dynamics to know whether reinfection was possible, as opposed to the coronaviruses that the children get. So I'm hoping it's more like SARS-1, that if you're sick, you develop both a T and a B cell response that will be protective in the future.
Yeah.
Well, this Queens community, which is Corona.
yes, that's
it
classic.
Is Corona, yeah.
How about that? It would be interesting to see if they get reinfections in the next month, right?
Yeah, I mean the nice thing about the serology testing, right? A lot of people poo-pooed it. Why are we doing this? What are we going to do with it? I said, you know what? We don't know what we're going to do with it now. It's almost like the smartphone when it first came out. We don't use it for phone calls, but we are going to be able to get a sense of people who were infected, who had positive serologies, titers at whatever. We'll know a year from now very much, what reinfection rates are, what happens, because right now we're saying, to really know in such a short window, you need sequence, you need that original virus, you need the second virus. But when numbers go up again in New York, we're going to see, are we readmitting, the same person we readmitted last year?
Right.
Let's hope that this doesn't happen with vaccine immunity.
Yeah, my hope is it doesn't. People take what I say and they say, oh my gosh, this means the vaccines won't work. And I'm not saying that, actually. I think that, this is about the virus's ability to subvert a proper, durable immune response. So if you use, a Moderna, if you use one of these synthetic vaccines, it makes me worry a bit about attenuated viral vaccines because, maybe you just leave in there the parts that prevent long-lasting immunity.
I mean, an inactivated vaccine should be okay also, right?
Yeah, it should. It shouldn't be producing whatever proteins are triggering this.
I'd like to know what that protein is. I'm sure you would too.
Yes.
All right, 2 emails today, or 2, 3-ish. Thomas writes, I'm an adult cardiologist in Philadelphia with a 4-year-old son. February this year, he tested positive for influenza A. 3 days later, after persistent fevers, started on cephalosporin for bilateral otitis media, completed a 10-day course. Then about 4 days later, around the last week of February, developed a terrifying constellation of symptoms I'd never seen in him before. Not being a pediatrician, his hands, feet, knees, wrists, elbows became swollen, red, and painful. He could not walk, fit into shoes, complained of a headache and stomachache, broke out in a rash, spiked a fever. These persisted a week. COVID was supposedly not yet in Pennsylvania, and the pediatric ED diagnosed him with serum sickness from the cephalosporin. But I wonder if he actually had COVID, and that was multisystem inflammatory syndrome. Now that we know that this coronavirus was present in the population much earlier than previous thought, what is the likelihood it was present in Pennsylvania early in February? Is the way to determine whether strange cases like this actually represent MISC. Is the antibody test of any use? And what are the recommendations for following children who have had MISC? Do they need to see a pediatric cardiologist, pulmonologist, rheumatologist? Kids survive, but for some of them, this is just the beginning. Thank you for sharing your knowledge and brilliance with the world. We need so much more of it. He means you, Daniel.
Oh, I'm sure he's referring to everyone, but thank you. In cases like this, and if I actually look at the original UK analysis, thinking things like serology, because by the time you have symptoms that might be the multi-inflammatory syndrome of children, it might be hard to detect the virus. So maybe in a case like this, because we do think it's antibody mediated, right? It's being treated with IVIG and steroids. So serology is potentially an avenue to pursue to try to see if that might be what happened here. And again, in people that have that, I would suspect that those antibodies are going to persist longer. We do know now that this virus has been circulating in the tri-state area for longer than we realized. Cases definitely in mid-February, and serologies were already positive in mid-February, which means people were getting infected in January in the tri-state area. So, the timing doesn't throw me on this. As far as follow-up, right? A lot of us are extrapolating from Kawasaki's disease. So yeah, these kids should get regular follow-up, but we're extrapolating. So, hopefully that will be sorted out. We have hundreds, right, of confirmed cases in the New York area. So it might make sense for a child like this to try to go to one of those centers where they have enough individuals that they're going to have an experience.
All right. Iwa writes, I'm a pharmacist and medical journalist from Sweden. I regularly listen to your pod and usually write a summary of Daniel Griffin's part for the physicians that are too busy to listen for themselves. I was a bit intrigued by some data from yesterday. That was last week, I guess. If I understood correctly, Daniel said that serology starts trending down, and at about 6 minutes in, if you look at people, this is a quote, if you look at people 2 months out, everybody's antibody levels are trending downward. And if you look at people with more milder disease, maybe 40% are now serology positive. People who were sick enough to have high antibody titers, you start to see them turn serology negative. And then she gives a link to a Nature paper which says 40% of symptomatic individuals became seronegative and 12.9% of the symptomatic group became negative for IgG in the early convalescent phase. And she writes, I just want to make sure that I understood and pass it on correctly. Could you help me find out if Daniel has another source or if he meant to say serology negative? I also want to send my warmest thanks for a great pod and that we really appreciate Daniel's updates. You are all great.
So yes, it was serology. She got it correct. I misspoke. So 40% of the asymptomatic become seronegative.
Got it.
And it's actually, so it's a good paper. I mean, it's this paper in Nature Medicine, and a lot of what bothered me, what a lot of the basis of my concerns were figure 3 of that paper, where they look at the proportion that are IgG negative, during the convalescent phase. And those are the numbers. So 60% are IgG positive. These are the asymptomatic, we'll call them mild. 40% are IgG negative. And then it's a lower percent are losing their antibodies. But if you look at the slope of everyone, asymptomatic and symptomatic, the slope is going down for both groups.
Okay. And finally, from Richard, dear TWivers, ivermectin was addressed in an earlier TWiV. It's apparently being used extensively in South America and Asia. Please comment and ask Dr. Griffin if he has any take on use of this medicine for C19.
I expanding which hospitals I go to. Chuck, I don't work enough, right? So I wanted to add another hospital that I could visit to my day. And so I was over at St. Joseph's. It's one of the Catholic health system hospitals here in the New York area. And as I'm coming new on staff, you meet with the credentialing committee. And actually one of the people in that committee was like, okay, so let me ask you about ivermectin. And the story was he knew someone who was in South America who got ivermectin and they got better. So obviously it was the ivermectin. So Vincent, I discussed your wife and Campbell and the fact that if you took 10,000 pills, you could, or actually be 50,000 pills, right, to get to the therapeutic level of ivermectin that was shown in the cell culture. And that paper, I hate the title of that paper. The FDA approved medicine ivermectin, is effective for SARS-CoV-2 because it makes it sound like it's FDA approved for COVID-19 treatment. Because people all the time say, "Oh, but it's FDA approved." I'm like, "Well, yeah, not for that." But it is interesting if you look at some of the physician experiences down in South America. They've been using it quite a bit, but again, it's outside of randomized controlled trials. So hey, the people we gave it to who could take oral ivermectin did better than the people who we decided not to give it to. So. I'm not sure there's a lot of, in my mind, science that explains to me why it should work. But it looks like there are going to be some trials. So we will probably get some data on ivermectin.
Okay. By the way, St. Joseph's, is that in Patterson, New Jersey?
There's a bunch of St. Joseph's all over the place, but this is St. Joseph's on Long Island.
Long Island. Okay. Because my dad worked at a St. Joe's in Patterson. That's why. And it would be funny if you were there.
Yes.
All right. Daniel, thank you again. Have a great week.
Pleasure as always.
And Chuck, thank you for joining us.
Good to see you both. Be well, Daniel.
Thank you.
Joining me today from Fort Lee, New Jersey, Dixon Despommier.
Hello, Vincent. And it's a miserable day out there today, as you know, because your window faces me and I face you. And it's Tropical Storm Faye moving up the coast of the Atlantic coast of the United States, dumping huge amounts of water wherever it goes.
Yeah.
So we could use the water, but not all at once, unfortunately.
Can barely see out the window here.
Yeah, exactly.
Also joining us from Western Massachusetts, Alan Dove.
Good to be here. And it is heavy overcast with an 80% chance of Faye this afternoon.
It's going to hit you too as well?
Yeah. They're saying inch or two of rain. We've got a flash flood watch in effect now, and so. All
right. Well, I can tell you, our next guest, it's not hitting him, from Austin, Texas, Rich Condit.
That's correct. It is not hitting us. Hi, everybody. It is 93, headed for 98. And then as of tomorrow, we have a string of 1, 2, 3, 4, 5 days where the forecast temperatures are 100 or more.
There you go.
It is sunny, so welcome to Texas. And I would also report that the 7-day moving average for hospital admissions for the Austin metropolitan statistical area is now at 71.6, down just a little bit from yesterday, but still in the red zone. We're hanging in there. I hope to see it drop, but we'll see.
You got mandatory mask wearing, don't you?
The governor has mandated mask wearing. I don't get out enough to know how much attention people are paying to that.
Probably not much.
Well, we went and voted today. Tell you, everybody there was wearing a mask.
Good. Very good. Well, we have a guest today. She is from the Icahn School of Medicine at Mount Sinai, my alma mater. Viviana Simon, welcome to TWiV.
Hello, everyone. Excited to be part of this.
You were in the same department where we were saying I got my PhD.
Yeah.
And your chairman is Peter Palese.
About eight or 9 years ago, Vincent?
It was actually 1970. What year? 1979 I finished my PhD. Oh, it's so long ago. My gosh. Now, Viviana, you said just now you were not allowed to leave Manhattan. Why is that?
Because we have to work so hard.
Oh, I see. Okay.
we have to shelter in place in the labs.
I see. Oh, no, it had nothing to do with quarantine. Okay, got it.
Show us your ball and chain.
Okay. So you are also in the Department of Medicine. You're a doctor, right?
Yeah. I am a European trained MD. So I'm not, I can't really see patients here. So I run observational research studies, and we've been really busy over the past 4 or 5 months enrolling patients with suspected or confirmed COVID throughout the hospital.
So you tell us just a bit of your history before we talk about COVID. You say European. Where are you from?
I'm born in Argentina. I grew up in Cameroon and Togo. I went to med school in Germany, France, and Finland.
Wow.
I worked as a MD as an infectious disease and internist in Berlin, Germany.
Huh.
And then I came to New York City for 2 years to do a postdoc with David Ho.
Mm-hmm.
And those 2 years are now, 20 years ago. I never found my way home.
And
New York
is home. New York is home.
That's
now,
the kind of travel history of people whose parents were in the foreign service or the military. Is that right?
That's correct.
No, there's a third option.
What's the third option?
People who care about literature and public education.
There's a fourth one too. They're being chased by the law, but that we won't go into that.
They're also missionaries, I should imagine. But okay, so your parents went and traveled and taught.
Yeah. They worked for NGOs and did a lot of publishing books and building publishing houses for better.
Very cool.
Fiction.
Interesting.
So
is your birthplace Buenos Aires or some other place?
Buenos Aires.
Buenos Aires.
So
beautiful city.
You must have picked up your accent in Europe, right?
What accent?
Okay. You're right. Anyway, David Ho is a few floors below me now, right?
Yes, I heard that with great interest. Yeah.
His whole group moved here, and Steve Goff used to be right next to me. He moved down, so now the floor is empty.
Yeah.
As well.
I look forward to visiting once we are allowed to leave our buildings.
Probably next year.
Maybe.
So it's interesting. Cameroon. No wonder you worked on HIV, right? Because it started in Cameroon.
Yeah, so probably somewhere in the south of Cameroon, like 150 years ago. At the time when I was there, that was the late mid 70s, late 70s. HIV wasn't really a thing yet.
Right. So when you came to Sinai, you mainly worked on HIV, right?
Yeah, and my postdoc with David Ho and Marty Markowitz was all on HIV and transmission of drug-resistant viruses and restriction factors. And when I joined Mount Sinai, I had a lab on the molecular aspects of viral host interactions, but in the context of HIV.
How long have you been at Sinai?
Since 2006.
Okay.
Now,
14 years.
Is your lab all COVID, SARS-CoV-2 now, or is there still some HIV people? I noticed you just published a paper in early this year on HIV.
Yeah, so up to March 2nd, we had HIV work.
Mm.
And starting March 3rd, all HIV stopped, and we completely retooled everything to work on COVID. I have to admit that I had, for the past 2 years, developed a program that allows us to enroll patients with viral infection in the hospital. So we had, because I have many of my colleagues are leaders in influenza virology, and in discussions with them, it became apparent that there is a big risk for a pandemic, and probably a respiratory virus that would, with pandemic potential, would cause a public health crisis. And New York, as a very international hub, would be a place to see it early. So we developed this program with the idea to have access in real time to samples from clinical people. So I have IRB protocols in place. I have a close relationship with the Pathogen Surveillance program, where we work closely with the molecular lab from the clinical point of view. So we had all of this, and we had been following for years what kind of influenza strains were found in New York, and what diversity, and all those things from a surveillance perspective we had been doing in parallel to the HIV work.
It's almost as if people have been warning about this kind of event for years.
Yes. So this is not, who knew, right?
Yeah.
Who knew that this could happen?
Everybody here and listening, yeah.
So this is the Pathogen Surveillance Program? Is that what you're referring to?
So Mount Sinai has two programs. We have an pathogen surveillance program that is basically monitoring bacteria and viruses that cause disease in our patients. Harm van Bakkel is the director, and Mia Sordillo from the clinical microlab and us from microbiology contribute to that program. And it looks for, hospital-acquired infection. By doing precision surveillance, by sequencing the bacteria and viruses, we can look for transmission chains. We can help hospital epidemiologists to lock down outbreaks before they happen, or before they become too big. In the context of influenza, we looked at where do the flu strains that cause disease in New York, where do they come from? Surprise, they come out from Europe. So New York, from a influenza point of view, looks very European.
So how did you?
That's something we saw also for SARS. We basically retooled our program from respiratory viruses in general and flu in specific to become fully SARS-CoV-2.
So was that like a programmatic decision, or was it your idea? How did that work?
Oh, that was the team decided that, when we saw in January, we heard about this new virus, respiratory virus in China. Of course, we started preparing for sequencing and viral load measurements. My colleague Florian Kramer started working on ELISA assays. So we are a virology department, and we felt like this is our duty to prepare for this.
Yeah.
Also, we were, the opinions were divided whether or not in early January, opinions were divided whether this would be a big public health crisis or nothing.
Yeah.
So what you have there is really, it's almost like a miniature CDC. You're looking for the next epidemic and doing surveillance and just kind of keeping track of all this.
Yeah, but we focused on Mount Sinai patients. So I now dream of a citywide program where we could, be more proactive and actually look for the unknown. So
Alan, hopefully they're doing a better job than CDC has been doing.
They, from what I'm seeing in the publications, I would say so, yes.
Now, one of the things you've published on and focused on is serological assays, right? And so why did you, And then you have genomics as well. But is serological assay part of the pathogen surveillance program?
The serological assays, Florian Kramer is the master of all things proteins and ELISAs. So, and he's located, 2 floors down, and is Peter Palesi's postdoc number, I don't know.
Right.
350. And he developed the ELISA. He needed then samples from people that are seronegative, and those from people that was documented.
I see.
COVID. So to validate the assay, and because I had been collecting samples from people over the past 4 years, I could provide the necessary controls to actually validate the ELISA.
Okay, got it.
And so that's how this collaboration came.
Could we ask you some questions about serology?
Of course. I'm quite knowledgeable, to my own surprise.
So you developed an ELISA assay, correct?
Dr. Florian Kramer did.
Yeah. And so this is an assay that needs to be done in a laboratory. You can't do it in the office, point of care, right?
No. So it's not one of those.
Not a dipstick?
No. This is a real, a 2-step ELISA. There is a first screening step using the receptor binding protein from SARS-CoV-2, which is done at a single dilution of serum to just see yes and no.
I see.
And if the sample is positive, there is a second step that is done with the full-length spike, where we do a titration to get an endpoint.
So this gives you a much more quantitative answer than something like a dipstick test.
Much better. It's specific. It's sensitive. It has emergency authorization by the New York State and the FDA. It's actually the only assay that comes out of an academic center that got emergency authorization from FDA.
Oh wow!
By doing the first step with a screen against the receptor binding domain, do you miss some sera that might be not positive to the receptor binding domain, but have antibodies to epitopes elsewhere in the spike protein?
In principle, yes. In our hands, we think the vast majority of samples detects the receptor binding domain.
Okay.
Some samples have. We think the receptor binding is one of the main targets for immunogenicity. There might be some samples where we have not seen any samples.
Okay.
Except maybe some people, some patients that have unusual immune systems, be it because of cancer, because of chemotherapy, or some kind of primary immunodeficiencies. I could imagine that there is some unusual antibody patterns to be seen. But there's someone with an competent immune system makes antibodies against the receptor binding domain. And in general, we see higher responses for the spike, which would be in line with having additional epitopes throughout the spike protein.
Is anybody else using your assay besides Mount Sinai that you've developed?
So basically, there's a very exciting thing over the past 4 months was that things that normally are impossible all of a sudden became possible. So one was that Florian Kramer's ELISA could move within a week, really, from the research space into a clinical, clear, certified lab space. So this allowed Mount Sinai to basically start testing volunteers for antibodies starting mid-March. So because as a research lab, we are not allowed to provide any clinical diagnostics. Right. So that all, that is, maddening to know that you have a way of measuring whether or not someone is SARS-CoV-2 positive, because it's a simple PCR. All of us can do that, but you're not allowed to do it. So that's where the magic happened here at Mount Sinai. where thanks to the leadership of the hospital, those things all of a sudden became possible. And so the clinical microlab had all of a sudden one of the first antibody ELISAs that could measure how much IgG was in someone's sample. And with that information, Mount Sinai started a antibody-rich plasma program really quickly.
And so
that was at the time the only thing one could do.
So have other high complexity CLIA certified labs also adopted this test, or is it?
Yeah, so the New York State is doing something similar. The Assays FDA approved. A lot of different companies have licenses. Florian has sent out the reagents to 250 labs all out over the world. There are detailed cooking recipes so that anyone can repeat this.
Right.
So
what is the what's the correlation between ELISA positivity and neutralization?
Yeah, that's so neutralization. There are lots of different assays to do it. Florian's lab has looked at neutralization using the full virus, and there seems to be a really good a correlation between antibody titers above 1 to 960. Everyone neutralizes with good efficiency. With lower antibodies, there is less of a.
I see.
But there seems to be a very good correlation between how high a titer is to neutralization.
Have you
noticed any
neutralization in viral cells? Whether neutralization in viral cells translate into protection.
I'm glad you said that. Yeah.
Real question people actually ask if they talk about neutralization.
Yeah.
They don't really mean neutralization. They mean, are we protected? Are we
Will
safe?
it neutralize it in me? Yes.
Yeah, Am I safe? That's the question. Am I safe? Do I have to worry? And that's truly a question we can't answer in a BSL suite.
Right. Is there a correlation between disease severity and antibody titer?
No.
No. Okay.
So the more severe,
the more severe the disease, the higher the antibodies.
Okay.
It's quite. Yeah. So it seems like a lot of virus equals a lot of antibodies.
So there is a correlation between the severity of the disease and higher titers of antibodies.
Mm-hmm.
Okay.
And back to the epitopes. Sorry, Dixon.
That's okay.
You can be next,
I'm not a
Dixon. virologist.
Why acknowledge it?
You're a virologist, Dixon.
It's probably a really good question. We'll get to it.
Do you have any sense for how many epitopes there are on the? No. Okay.
No, not yet. Not yet. I think all of this, we have distributed all our samples to our different collaborators, and everyone is trying to piece together what epitopes, which are the most important outside of the receptor binding domain.
When you distribute reagents, does that come along with positive controls?
If you ask me nicely, yes.
So my question is, do you know if anyone is longitudinally studying patients using your ELISA test to see how far out in their recovery the antibody titers last before you can no longer detect
So
them?
we do this. We have an NIH-funded study called Paris, where we follow healthcare workers over time to see how long the antibodies last. And whether or not people get reinfected. So we have two arms, one for the people that have already experienced and survived COVID, and the other arm of the study is for people that are seronegative, and which will allow us to look for the attack rate of the infection by looking at de novo infections.
Is it too early to tell us maybe some early results?
So what we see is that the antibodies we are now in month 3 of the study, and so far what we see is that the antibody titers remain quite stable with a very slow decrease. But it's not. There have been a couple of studies out on preprint servers as well as in print already, suggesting that those antibody responses are very short-lived. So that's not something that I see.
Now, how about people who I assume you're correlating infection by a PCR test with antibody titers by ELISA. do you see people who have definitively had the virus but don't generate antibodies?
No.
Great
Okay.
question.
No. And we have, because I work closely with the director of the plasma screening program. So Mount Sinai has screened over 25,000 people for this plasma donation program. So we have antibody data of 20,000 people. And also when people had a documented molecular test showing that they had SARS-CoV-2, 99% had also antibodies. There might be 1% of people that have clear documented SARS-CoV-2 that don't make antibodies in the classical fashion.
So in that study, what's the earliest in the year that you detected a positive COVID-2?
So that's it's out in a preprint. We did a serosurvey by looking at samples from our hospital that were collected from patients seeking care, and we had access to plasma samples going back to the middle of February.
Okay.
And so middle of February, we could see that the first seropositive samples appeared, and then the prevalence slowly but surely increased.
Thank you.
It's a preprint that is currently undergoing.
So it's on medRxiv, right? I think.
Exactly.
So what's the overall seropositivity in New York City?
That depends. I think, like, often you need to look probably in an average between around 5 to 10%, but in different populations it will be much higher.
Yeah,
and in others much lower.
Yeah, there's a wonderful map in the New York City Department of Health showing it by, what is it? What are the little
zip codes?
Zip codes, yes.
Yeah, zip codes.
The highest is Corona, Queens. How about that?
Really?
Where else would it go?
68%.
Yeah.
What do you think about that? Is that real, you think?
Yeah, I think so. So some communities in New York City were very hit hard and early.
Yeah.
And those are, tight-knit communities. Unfortunately,
might also have been some super spreader events in there.
Maybe. So we also did a lot of sequence analysis of the early strains in New York City that we got from patients in early March. And by looking at the sequence of those viruses, we could basically reconstruct a time tree. And this, so the diversity of the strains suggested that there were multiple introductions into New York City, followed by then community transmission that went untracked for a couple of weeks.
Wow.
And the
and those communities could have well been affected by those untracked forward transmissions,
right? That
spread without really getting the attention needed because there was no molecular testing available in end of February, early March.
Where was the first case in New York City? Do you know?
Yeah, I know very well. It was February 29, and it was at Mount Sinai.
Mount Sinai. It was
and we have grown the virus, we have sequenced the virus, and we have deposited the virus for the community to use.
And did that come from Europe, that particular sequence?
No, that was a travel-associated infection. It came from the Middle East.
Oh, interesting.
So our New Jersey's first case was right where I'm sitting in Fort Lee, and it was a Korean that came back from Korea with the virus. Yeah.
So February are the earliest cases you think in New York City then, right?
Based on the serology, we think, and the sequencing models, I think it probably circulated in New York City early February.
I think in the paper you published, you also said there were some introductions from Washington as well, right?
Yeah, Washington State.
Yeah.
Oh.
Yes.
So does your data set pick up asymptomatic or pre-symptomatic infections?
No, because our source of samples are people that seek care at Mount Sinai.
Right.
I think we are enriching for symptomatic people because they come at least to the emergency room. So the asymptomatic patients it's a difficult topic. We start so I have those really broad study protocols. So people reach out to us if they have unusual presentations. And over the past couple of 2 weeks, I have received a lot of calls from physicians that observed possible reinfections. So those are sometimes individuals that are completely asymptomatic and get tested as part of some tracking contact tracing programs because they have been in contact with someone that tested positive, and then everyone who was at the dinner party gets tested, and all of a sudden they turn out to be positive, which they never expected to be because they have high levels of antibodies. So that's currently what we are working intensely on. To understand, what kind of antibodies allow reinfection. Is it actually a real thing, reinfection, or is it just more a shedding of dead viruses? All of this is still possibilities. We do not really know if people got really reinfected with a different virus.
Yeah, no one has sequenced the first and the second virus to see, right?
Yep. Yeah. Exactly. So that would be the real test to have the first virus from March and then now have the second virus from June.
Yeah, because, we never thought Ebola persisted in people, and it turned out it persisted for 3 months, and it could recrudesce right out of the eye and other places. So
this could be something similar happening here, that one, just keeps shedding virus. Again, experimental evidence suggests that if the viral load is low, the virus normally is not infectious in vero cells.
So you were saying you were testing serum to identify who might have immune plasma. Were you testing?
My colleagues. It's a big we. It's a we.
Did you run trials, clinical trials for convalescent serum to see if it had any effect on the outcome of the infection?
Again, not me, Mount Sinai, my colleagues.
Yeah, no, I meant you, Mount Sinai.
we mean y'all.
Y'all, that's right.
Y'all.
Authorial you.
This was a gigantic team effort here at Mount Sinai. And Mount Sinai has started a antibody-rich plasma transfer program starting mid-March, really, really early, which then initially it was just us requesting permission from FDA for every patient.
Sure.
And eventually we joined forces with a Mayo clinic that had a multi-center site study for plasma. I think Mayo and Hopkins, those were the big multi-center studies to look for plasma transfer, its efficacy. Some studies have a placebo arm because that's truly what you need if you want to determine whether or not there is an effect. So when we started here out at Mount Sinai, we did not have a placebo arm. We decided that it's a public health emergency, and we try to help in this crisis. It was truly, truly daunting. The early weeks of March here, it was truly overwhelming. So we had, the hospital needed extra beds. So there were tents in front of in Central Park. It was a very scary time.
Right.
And since there was very little to be offered to patients, this antibody-rich plasma was a lifesaver.
What are the results of those plasma therapy studies?
Modest effect, I think.
Okay.
Especially, so I'm only aware of the early studies where it does has a positive effect, but it's not a silver bullet. But at the time, the serum the antibody-rich plasma we got was from people that were maybe 2 weeks out of the infection. So now with the quality of the antibody-rich plasma that is available, is much, much better because the antibody titers are orders of magnitude higher. Because people, our immune system needs time to produce good antibodies.
Yes.
Like so many things, there is a bit of maturation involved.
Yeah.
So, and titers need to be mounted. So I think the later studies are done with much better antibody-rich plasma, with higher titers, and this could influence outcome of those studies.
Seems to me that if if you're just giving that to people in the hospital who are very sick, it's probably where the virus titers are already declining, and it's not going to have much of an effect, right?
Yeah, that in addition was the case that in SARS-CoV-2 infection, it seems that the virus is only one of the determinants of disease severity, whereas then immune dysregulation and dysregulation of coagulation takes over after a certain point.
Right, and a lot of the as you've seen in your sero studies, a lot of the very sick patients are producing high titers of antibody.
Yeah.
So it's not a lack of antibody that's necessarily holding them back.
Yeah. All this knowledge didn't exist when we started doing
right.
Sure.
This is really kind of
Well, now there are now there are human monoclonals being tested, and they're going to have the same issue. You have to give them earlier if you want to have. So I would guess you could give them to frontline workers, healthcare workers, and so forth. But since they have to be given intravenously, right? They're not going to be able. You can't take it at home.
No. But I think they're about to start some trials of it, both as therapeutic and as prophylactic.
Yeah.
Right.
Okay. So I wonder if you could talk a little bit about the sensitivity and specificity, the positive and negative predictive values. Could you explain those in a way that we can understand or try?
So this is those measures are basically attempts to predict how often one is wrong.
Mm-hmm.
So with a diagnostic assay, and that's true for any diagnostic test, there's a trade-off between how sensitive a test is, how many how many infections will be picked up, versus specificity means how often will the test be right. And that's the positive and negative predictive values. So of course, you would want your antibody assay to be always correct, and you want it to pick up every single person with antibodies, and that's not how it works. So the assays are either sensitive, and the negative consequence that some of the positive results will be wrong. So it's called false positive.
Mm-hmm.
Otherwise, a test can be very specific, but the payoff will be that one misses some of the infections. So that was it's a very important notion, because at one point in mid-April, we were discussing whether it would make sense to test people for antibodies and then provide some immunity passports to people that have protection, which was inferred. Still we don't know if antibodies really protect or not. This is matter that studies will show, like the studies we are doing here at Mount Sinai. But the false positive rate is very, very concerning, because people would think they have antibodies, but in reality they are naive. So that's and even a really small, even if your assay is 98% specific, that still leaves you with 2% false positive rate, which translates into hundreds of thousands of people that will get a wrong answer if one uses the test in a population with very low risk of infection.
And since we don't know to what extent the antibodies are protective, we'd also be getting answers that could be misleading on a qualitative level, right?
Yeah. In addition, many of the antibody tests are really bad.
Yes.
So I'm sorry to be so blunt.
But not yours. Yours is very good, right?
Of course. Ours is the best.
So in terms of antibody tests being bad, can you comment on the point-of-care tests that we see out there, the lateral flow assays, and whether any of those, to what extent they meet the need, put it that way?
Very, very wide range of quality. Some very good ones and some really bad ones.
And how could one, is there somebody keeping track of this and saying, this is good, this is bad? I mean, presumably the FDA would, right?
Yeah, we would hope that.
We would hope that, wouldn't we? Yes.
That's not a resounding endorsement.
Still a swamp.
Because the that study that was done, some of the city serosurveys, the one that was reported with a 68% positivity in Corona, that was using, as I understand it, a point of care test that was being administered by City MD, one of these, I guess they're an emergency care type outlet.
Yeah.
So I suppose you could say on the one hand that the reliability of those results would depend to some extent on the test. On the other hand, 68% is a big number. So I would presume that there wouldn't be a test that would be so bad that 5% would get reported as 68%.
Probably not.
I hope not.
Well,
it depends on how big the kickback was to use that test.
Ooh,
I'm not accusing anybody. I don't.
What?
That
Viviana, what are the numbers for your assay? What's the sensitivity and specificity?
I think 99 point something specificity and 98 or vice versa sensitivity.
Now,
it's among the better ones.
Does it help to have the test twice? Does that improve the number? So if you get a false
our test is already two tests.
Right.
There is our test is this two-step process where there is first a screening and then a titration behind it. So that gives it the really good results.
Yeah, because we've had many people write in and say they were sick and they had 2 serological tests in a row and they're both negative and they're wondering. But that's possible, right?
Yeah. They didn't have COVID.
Well, they might not have
they
had COVID.
had flu. They had flu. They had para-influenza, RSV. There are lots of other respiratory infections that make us sick.
Yeah, I notice this all the time in publications that we've seen where they'll have reporting on 2 different sorts of populations of people. One is people who have reported symptoms, and the other is people who are PCR positive. And typically, the seropositivity is much, much higher in the people that are PCR positive and much, much significantly lower in the people that appear to be symptomatic. And that tells you something, I think, about what symptomatic means. There's, as you say, lots of respiratory infections out there.
Yeah.
But it's not impossible that a COVID infection would give 2 false negatives if the assay isn't very good, right?
And so you need to distinguish between the molecular tests that look for active viral replications that amplify the virus from the nose or the throat. So those are molecular tests that look for the virus. There it's possible to miss the virus because it might not be replicating efficiently in the nose of everyone. So we have seen false positives where a patient presented with symptoms really suggestive of COVID, but they tested repetitively negative for the virus. A couple of weeks later, they would have antibodies.
Got it. Okay. Do you know anything? Go ahead.
So the test, it's really important to not confuse the molecular test with the serological.
I was just talking about 2 serological assays in a row that are negative. It's possible depending on what the test is, right?
Yeah. Did this person test positive for the virus at one point?
I don't know. We have a lot of people writing who give us stories, Some of them are physicians, actually, but I just wondered if a test could be faulty enough to give you 2 antibody tests could give you 2 false negatives in a row. It's possible, I suppose.
Yeah, we have not seen that.
But
again, we use our assay.
So you mentioned the PCR tests. Some time ago, there was a suggestion that one could use saliva. Do you know anything about that?
Yeah, you can use saliva.
Yes. Is it as reliable as a nasopharyngeal swab?
So there are some studies, I think, out of Yale where they compared nasal swabs to saliva, and it looked very comparable, if not better, with saliva.
Okay, but my impression is that many places are not using saliva. They're still doing the swabbing, right?
That has something to do with the regulatory aspects of clear certified labs. So in order to use different materials, one needs to do a validation study in the lab. Because right now, all the molecular tests are basically have emergency approval for nasal pharyngeal swabs.
Okay.
And in order to test saliva, the labs need to jump through extra hoops to do that.
And if it ain't broke, don't fix it. I think there's a temptation to just stick with the nasal swabs because they do work, and
and it's a lot of work. Everyone, but no one has really the
additional approval.
Additional approval, additional submissions. No one has the bandwidth. Everyone tries to just try to keep up with what is needed.
By the way, what is when patients come to Mount Sinai? Where are they coming from? Just Manhattan or all the boroughs?
And so Mount Sinai it's a large healthcare system with 8 different hospitals in Manhattan, Brooklyn, Queens.
Oh, okay.
So
it's not just Manhattan. Got it.
It's not only Manhattan, and we have centralized labs. We get the nasal swabs from all the different hospital and care sites. That's where we kind of source our viral collection.
Okay.
From.
Now, right now, I just looked at the map for New York City. I mean, the infection, 118 was the last reported daily cases, very low hospitalizations, 4 deaths. So we're really, are you surprised that it's so low here? Because other places like where Rich is, it's going crazy.
Yeah, no, it's terrifying to see what happens in other states. And I'm very, very worried that it might come back to New York City.
So maybe this is a silly question, but what's the sort of citywide seropositivity? Maybe you've already said this. Maybe that's an irrelevant question because regional variation is going to make a huge difference.
Yeah, so we have large differences in the zip codes. Vince, you had the map in front of you. What is ranges from 60 to 0.5, depending on where you look.
And the darkest, which means the most cases, are Corona in Queens and at Bronx. Parts of Staten Island. But then if you look at Manhattan is all pretty few cases, especially all the way down in the financial district. There are very, very few cases.
Who lives there? Yeah.
Oh, man.
So it really varies. It's a wonderful map. Yeah. It's by zip code, as you say. Yeah. And you can do cases, hospitalizations, and deaths.
Because, what I was thinking was, in terms of the disease returning to New York, I was wondering whether, in some areas, they are going to experience some herd immunity.
Yeah, that's a good question.
I guess we'll find out.
Yeah, so far it's still, you need probably.
I hope we don't find out.
Yeah, you would need like more than 70 to 80% of the people having protective immunity.
One of the things in Manhattan's favor is that restaurants are still closed, right? You can't go inside, no theater, there's no bars. So in other places, they're allowed. So maybe that's helping, right?
Yeah, I'm sure that behavior is the key to all of this. And, and finding our way through this has to do with identifying what the really appropriate behaviors are in order to keep it under control without, dying for economic reasons.
And we have identified those behaviors. The Northeast, New England is doing great, and here Connecticut is, I think, now the lowest rates in the country. Massachusetts is way, way down. And in Massachusetts, very, very early in the pandemic, we had a super spreader event at a meeting in Boston, and it rocketed us way up numbers. Biogen, yeah. Not to pick on the company, but that did happen at their meeting, and that's now we got that down, and we got it down by doing all the stuff that all the public health people said we needed to do, and now we're very gradually reopening things and having discussions like, gee, do we really want to reopen the bars? We want to, but do we? Is that a good idea? And the consensus seems to be maybe not. And we did just reopen gyms, but the requirements for them. I got an email from our local community center where I used to go and work out 4 or 5 times a week, and I looked through the requirements and I said, "Oh man, I'm not bothering with that."
Yeah.
You got to reserve a time. There's no access to the locker rooms. There's just, you basically would go in, use one piece of equipment, and leave, and that's the kind of thing that keeps it from spreading.
So the museums actually have that as a requirement. You have to write ahead, get a ticket, show up on time. There's no refunds. You either go or you lose. So Texas, you have beaches, you have spring breaks, you have lots of
when they decided to reopen, the place just went nuts. It was as if nothing had ever happened.
That's exactly right.
That's a bit of an exaggeration, but
no, you're right.
They opened, although they had some occupancy limits. They opened the bars, they opened the restaurants,
right?
And then people misbehaved. There was no mask rule.
Nope.
And people misbehaved. They engaged in large gatherings, and it just went nuts. Okay, so it's really interesting. This is the influence of group behavior on on the outcomes.
That's right.
Viviana, by you on the street, are most people wearing masks?
Yes. I think because New York was such an early epicenter of the pandemic, everyone realized how incredibly scary this is and how quickly things go out of, spin out of control. I think that really made compliance so much greater, because everyone knew someone who had died, someone's mother or grandfather that passed away in incredibly quickly. And knew that the hospitals are, people are treated in tents on the street. And
I would say the tents on the street would get your attention.
The refrigerated trucks parked in the street probably had a big impact. Yeah.
Yes, I think they were really well camouflaged, so not everyone picked up on them. But it was really scary, and so that kind of motivates you to wear a face mask or to leave town. I think many people, people also, because one could work remotely. So Mount Sinai basically closed down its entire non-essential research operation, and everyone was encouraged to work from home. So all the labs that were not working on COVID closed down. So
that may have a lot to do with the very low seropositivity rates in Manhattan, where a lot of if you track that by income level, I'm sure there's a correlation. People who had someplace outside the city to go to did. And people who didn't, who are maybe, recently arrived or what have you, or they live in their local neighborhood in Corona, Queens, they don't have any place else to go. And so they stayed. And
or they have to work, right?
Or they have to work there. Yes, essential
work.
workers, right. Your blue-collar neighborhoods, your Staten Island. Yeah.
One of the ironies of that is that a lot of the New Yorkers tried to go to Florida to escape the coronavirus. So... you open one door and behind it, wait a minute, I thought I just saw you over here.
And I do believe in the early times, Florida really forced New Yorkers that would go to Florida on vacation.
Turn around. That's right.
Yeah, exactly.
Close the door.
I heard some anecdotes about that.
That was so silly. So silly.
Viviana, in one of your papers, you mentioned that there was a delay in testing in New York City. Can you tell us why that happened?
Initially, the CDC guidance was to only test people that fulfilled a certain set of criteria, which was basically a travel history to a region defined as risk, which was Wuhan, and in later time points, later in February and March, also Middle East and some regions of Europe. People could get access to tests. And the test would be basically, how do you say, allowed, supported by the Department of Health and the state labs.
I see.
So it was, access was restricted, and it took a really long time.
So the problem New York had initially was that they were listening to the CDC.
Well, there was no other approved tests. The only test available was the one through the state labs.
Yes.
Mount Sinai worked really hard again, transferring tests from the research lab into the clinical lab to basically run through the approval process of getting independent assays approved.
Right.
That requires a lot of work, a really, really well-organized pathology department that you can pull that off.
So that obviously will not happen in every state, and that was part of the problem, right?
Yeah, so all this diagnostic testing and regulatory process are very state dependent.
Yeah.
So New York City right now has no limitation of molecular tests, whereas this is still a problem down in Texas and other states where there is still a mandate for centralized testing. And then people have to wait like 5-6 days, which is really complicating any contact tracing. And, it doesn't allow people to protect their loved ones because they don't know that they're infected.
Boy, if we could only go back and change that early policy of the CDC, right? And get started testing earlier. We should have been testing in January, I think.
Of course, and we should have had good assays, good tests.
That's the other thing. The CDC.
I think you need to go back to 2016 to fix this.
The CDC's original, they were distributing the tests themselves, right? The test kits, and they were faulty, I think, right?
Yeah, I think there might have been a combination. There might be some primers that were less than specific, not optimally specific, and I think there might have been also a PCR kit that provided some.
The CDC insisted on developing their own test and not letting anybody else handle it, and there was a definite delay from an error in that process that put us back at least 2 weeks, and there's a chain of problems that you can look into.
But Viviana, New York, so Sinai decided they were going to do it themselves, right? Is that what I'm understanding?
So So Mount Sinai labs implemented a CDC-like assay in their own lab and went through all the procedures.
I see.
Emergency approval. And at the same time, we got also the molecular testing from companies that had their tests approved. So all in early mid-March when we could go live with testing. But it took 2 weeks really to get all of that on board.
All right. I'm going to let Viviana go because it's an hour now. I told her she could go in an hour.
How did that happen? Yeah, I want to know. I want to know what the big questions are in her mind. What the things are that you really want answers to now. What you're working on.
Oh, I have a lot of them. I need postdocs. I need lots of postdocs. I have lots of great ideas. So of course we are going to continue working on SARS-CoV-2. I look really forward to learning about what antibody titers are necessary for protective immunity. I think that's really interesting. I really, really hope that we will understand whether or not there are reinfections and under what circumstances reinfections are possible. But I'm also really interested in very molecular questions in the context of HIV. So we are studying RNA modification of viruses, RNA modification of HIV, but also respiratory viruses and SARS-CoV-2. That's super exciting. We have a project on CD4 stem cells where HIV persists. And we look at endogenous retroviruses. So I just hope that things will keep quieter and we can go back to some of the molecular studies. All the translational work is great, but I like also the really molecular basic work we are doing here.
So when do you think we'll have a vaccine?
A year, maybe, if we are lucky. So if this all is based on the assumption that antibodies protect.
That's right. Right. For most viruses, they do, right?
Like HIV, right?
Oh, yes.
Yes.
Well, most viruses.
Well, actually, for a vaccine to work, I mean, yes, the vaccine trials are looking at antibody production, but really what we care about is that the vaccine induces immunity.
Right.
And so if it turns out that the vaccine induces antibody and coincidentally it induces immunity by a totally different mechanism, it's still a win.
Still okay.
Yes, yes, absolutely.
That's why we do the clinical trials.
Yeah.
Right. Yeah. So what do you think about the schools opening again?
Yes.
Yeah, I'm concerned. Really concerned. And, it will require a lot of thinking and testing and retesting and...
What about the teachers? I know they're not worried about the students. They say students are young, they can survive this. This is great. What about the teachers?
Yeah, I'm not sure that it's true for the children, actually.
No, I agree with that. But assuming that even if that's true, the youngest person in the classroom is not the students, or rather the oldest person.
Yeah.
It's the teacher, and the teacher is taking a big risk every time they go into class.
Yep.
Speaking of testing, I mean, what we really need is a really accurate 15-minute nucleic acid test. Do you think that's going to happen?
Yeah, they will bring it down to an hour at least. I think we have rapid tests that will take
an hour. That's fine.
45 minutes, an hour.
And they
for flu.
And they will be accurate. And so you think they're going to be antigen-based tests?
Yeah, they will be antigen tests.
Yeah, that's what we need is a quick point of care test.
Not tomorrow, but those tests are in the making.
Got it. Okay, Viviana Simon, Icahn School of Medicine, Mount Sinai. Thank you so much for joining us.
Thank you for having me.
This was great.
Thank you.
Good luck. Stay safe, okay?
Thank you.
Bye.
Bye.
Bye-bye.
We do have some email here.
We do.
Just a few.
Gee, just a few. Yes, 135 pages at the moment.
Oh, we've cut it down quite a bit.
Those are the ones that are in the document, yes.
All right, so we have the first one is from what he writes from an upset Chinese listener. And I'm just going to say that in this email, the writer calls us names, and I normally wouldn't read it. So this is the last time I'm going to do this for this person or anybody else who calls us names. But I think we need to because I think the person is wrong and we're right. But we're not always right. But all right, dear TWiV team, I was looking for how American virology scholars were dealing with this pandemic, and the TWiV special with Ian Lipkin brought me here. I was listening to TWiV 67. At the very beginning, you were talking about how China approves Ad5 vector SARS-CoV-2 vaccine to only the military, and some of your comments really infuriate me. You said this is a phase 3 test, which I agree. But then you folks went on to say how it's like treating the soldiers as some kind of test subjects, bringing up how the U.S. military would follow CDC guidelines. I'm sorry, but this is just bigotry. Okay, that's calling us names. It's not bigotry. We're talking about science here. And if anyone calls me a bigot, again, I'm not reading their letter, okay? Then he continues, have you ever considered the possibility that we already have this thing under control domestically so we don't have a population to test the vaccine? Oh, that justifies it. Sure. Approving a non-efficacy tested vaccine. Perfect. Makes sense. Sorry. 2, global travel is rare and fully quarantined. 3, the military are basically the only people who have to make contact with foreign individuals in uncontrollable environments. I also follow opinions on our social network, virology-related students and professionals. Most of them have another angle, that the phase 2 probably proved that this thing's quite safe. Well, that's not the issue, bub. It's efficacy. And since we have Indians attacking us for nationalist reasons and we have some military overseas, them taking this virus is likely to be better than nothing while fulfilling some phase 3 testing purpose also. All right. Then he goes on, but I don't want to read the rest because you get the picture. And he says in the U.S. we have a systemic failure, which is absolutely right. Yeah. And he says, how could the scholars blame Trump when the entire society has been receiving false information? How are you supposed to train America's future generation of virologists when you are still bigoted even after you were part of the problem? Okay, so we're not bigoted. It's been approved without an efficacy trial. That's our problem. And you agree that it's basically a phase 3 trial. We wouldn't do that here. That's the point. And we don't have any information from you because we get very little. And so we're speculating. And we do that all the time. So we're not bigoted. You know how many times I've defended China that the virus didn't come from a lab? I go on China TV and do that. I never heard you once thank me for that. Okay, so don't call us names. If you don't like what we're saying, be nice about it. You can disagree, but that's my point. And Rich, you were involved in this. Do you have anything to say?
Well, a couple of things. I have to say that there have been times on the show, not necessarily that show, but there have been times on the show when we have made comments that I felt uncomfortable about, that were based on some potentially cultural stereotypes in situations where I personally, at least, didn't have sufficient experience with the culture so that I would be qualified to really comment on those stereotypes. And I think in particular, given our reach at this point, being global, we have to be mindful of that. And so, although I agree that the language from both directions has to be respectful, I think, I personally would like to be mindful of this. I think one of the phrases that came out that was probably particularly annoying in this case was, I think the term guinea pigs was used in the discussion of using a vaccine in the military. And that's probably an unfortunate description. Okay. Could have been perhaps a little more respectful in that case. I would say that relative to the possibilities, I think we did mention that we understood that it was very well controlled, under control domestically. And for that reason, it was going to be hard to find a good test population, and that would be justification for using the military. And that includes the global transportation. As I also recall saying specifically that we didn't know what sort of circulation the military had, so we didn't know what sort of exposure they were going to get. So we did consider those things and acknowledged when we didn't know exactly what was going on. I would also say, I save that. We had some comments about the U.S. military. There are some other sort of inaccuracies in this, and I don't want to belabor the point. I think, for me personally, I think a point's being made here that we have to be mutually respectful of cultural differences, okay, as we describe these things, and do the best we can to stick to the science.
Agreed.
Fine.
But don't call us names.
Don't, yeah. No, I was not on that show. That does not absolve me of blame. I'm sure I've said insensitive things on the show in the past. Don't call us names. But I would just add, and I've not listened to that discussion. I don't know if anybody brought up that the notion that the U.S. military would somehow be above this sort of thing is not necessarily based in historical accuracy.
Right.
I'm thinking of things as recent. I mean, in your generation, in the 1950s, we did nuclear tests, and we had troops standing, in the range of the bomb blast, and you could argue that we didn't know that was dangerous, but I think we did. And so there's not necessarily an ethical bright line that we can say that our military is not going to do this sort of thing.
I think what was brought up was whether they had a choice, right?
Yes, whether so.
And we don't know.
Yeah.
So the comment that I made on that show was that the U.S. military will not use drugs or vaccines that are not either approved or licensed by the FDA.
That is,
I didn't say anything about the CDC, but that's neither here nor there. I talked about the FDA. Now, the fact is that that's not strictly true either. I have discovered since then. I've had some correspondence with Ed Niles, actually, who brought up the anthrax vaccine situation, which I don't want to go into detail, but it's complicated. And there have been, put it this way, there have been circumstances where vaccines or drugs have been used in the military, and there's a lot of controversy back and forth about whether this sort of thing can be mandated, whether there should be informed consent. Okay, so there's not, as you say, Alan, a bright line.
And ethics, when you're dealing with a military force, gets complicated because the whole risk assessment, and this writer even mentions that. I mean, they're sending people into harm's way, and you can quibble about whether they should be sent there. But the point is, these people are in the Army, and they're being sent to places where what's the greater risk, getting this vaccine or not getting that vaccine? So there are points to be made on this.
Every country can do what they want.
Yes.
All we said was, this is basically a phase 3 trial, and he agrees. Why do you say it's licensed? I think that's like one-upmanship almost. Just say it's a phase 3 trial, because they're saying it's licensed. And maybe that was actually a miscommunication by the news. Who knows? It could maybe China does say it's a phase 3 and someone picked up that it was approved. Maybe this is all nonsense. But I don't see licensing a vaccine without a phase 3, but maybe China wants to do it, and it's up to them. And that's fine.
The other comment I have on this letter is that he points out our own cultural inadequacies, and as you already said, Vincent, I'm totally on board with that.
I think we've been very, very upfront.
Yeah, we've been hard on ourselves,
probably longer than I should have, harshing the CDC in our interview with our guest. And
the one of the most interesting things about the pandemic to me is that it has really highlighted many of our own cultural and societal inadequacies that I hope
for sure.
I hope those highlights don't go away, and that over time.
We admitted that we didn't foresee this for sure, and we admit that the country has blown it on many levels, no question. If you listen to TWiV and sounds like you haven't, we've talked about this a lot. But anyway,
we've actually specifically called China out for their excellent transparency early on. I mean, 2 weeks into this, they're providing sequence information, and the scientific collaboration has been great. So this is not a bigotry thing.
Dude, I've gone on many Chinese TV programs and confirmed that this virus came from nature. That's because that's what happened, in fact. I didn't have to do that, but I did because it was the right thing to do. So don't call us names and say that we don't know what we're doing and we don't know how to train the future generation of virologists, blah, blah, blah. Okay? That's it. Dixon, can you take Corvin?
Grumpy has spoken. I'm sure. Now I'll take the next one. Corvin writes, "Hi, TWiV team. I wonder if you've seen Yale's reopening plan. It seems pretty good. I looked at it. It does seem pretty thorough, and they're spending the money to test.
Testing twice a week. All entry students are being tested.
Every single student
and
is
twice
being
a week.
Tested
and this is Yale. They have a lot of students like Cornell, right?
That's right.
Can be done.
Yale also has an amazing student health plan.
Yeah,
it's true. So they started off decades ago with a great structure in place.
Yep. Remember, Cornell's medical school is located in New York City, whereas Cornell University is located in Ithaca, New York. With Yale, the medical school and the University and Yale University are in the same place. So maybe that has something to do with it.
Well, that sort of healthcare structure ought to exist everywhere.
Yes, I agree.
This is one of the deficiencies in our society that's been exposed by the pandemic.
No question.
Is the unevenness and inadequacies of the healthcare system.
Well, the thing that I'm upset most about with regards to that is that it's up to each governor. It's up to each mayor of each city. Since when did they become health experts?
And one of the worst parts about that is you got the states competing against each other.
Absolutely.
For supplies and stuff. That's crazy.
Come on down to Florida. Everything is now open. Or come on down to, I won't say Texas because you're there, but California says the same thing. Well, they all know they made a mistake.
They're competing with each other for supplies and stuff.
Yeah, you bet.
It's crazy.
Yeah, it's
crazy.
It's all crazy.
Hey, Rich, can you take the next two? They're short.
Okay. This is on the haiku arc. This is great. We're getting haikus. Peter writes, "Butterfly alights, Twivian wings gentle dance, heals virus-worn earth."
Isn't that nice?
Great. You guys keep me sane. God bless. Peter from Miami. And Nicholas writes, "The people who like the finer division of Fahrenheit have a point. So my proposal is this: just multiply Celsius by 2. Okay. Well, what would we have to do? The boiling point of water would be 200 degrees, right?
Yes.
But frankly, when I do I do a kind of conversion in my head where that's one of the things that I do. There's a factor of 2 in there somewhere.
Right.
Plus and minus 32 degrees.
Alan.
Mary writes, "Dear Twivers, I write to you from the Detroit suburbs where the effects of climate change are apparent as it is 92 Fahrenheit at 2 p.m. today, and we're having a week of nearly record high temps in the mid-90s. This is about 10 degrees above average. In response to Anonymous's letter in episode 629 regarding what explains the outbreak in hot and dry Arizona, you failed to consider one option. Perhaps it is the case that we humans are protecting the virus from the hot and dry weather. When the virus is inside a human, perhaps someone who brought it to Arizona from another cooler state, it only needs to endure approximately 98.6 Fahrenheit while the human is asymptomatic. When that human is outside, the virus stays comfy in its 98.6 Fahrenheit home. So any virus expelled through breathing, etc., might die in the heat and dryness. However, when that human is indoors, the temperature is likely much cooler, and any virus escaping will do just fine, especially if it's lucky enough to find its way into another human, and so it goes. This puts me in mind of another letter you recently read inquiring whether air conditioning might increase transmission somehow. It might be the case that what causes humans to use air conditioning, hot weather, also causes them to spend more time indoors, and it is that increased time indoors that results in more cases. Love your show, especially the banter. It is the banter of creative, nerdy types. I was turned onto your show by another creative nerd, Mike Pesca of the Gist podcast fame. Long live TWiV. And Kathy had a note on this one, which I'll go ahead and read since she's not here. She said, my friend Carol in Tucson was evidently shouting at the podcast that, quote, people go inside in the summer in Tucson. The virus doesn't have to survive in the outdoor heat.
But I mean, when people go inside, this increases transmission. That's why winter seasonal infections increase, because people go inside, at least partly. But if you're outside and you, as she said it, and you exhale it, it's less likely to be transmitted. Well, we know for this virus, most of the transmission is happening inside, right?
And who in Arizona is going outside in the heat of the day is the point.
I just
Mad Dog's an Englishman.
My wife and I were just having some correspondence with some friends recently, where it's another sort of one of these classic situations of a family getting together for a gathering, and they've all turned positive. You know, and nobody was obviously symptomatic when they all got together. Getting together with people for some reasonable period of time, you know, a few hours or something indoors, is a hazardous activity.
Also.
So the next one is from Jim. Hello, Twivnix, a loverly 92F 33C 552R 306K here in St. Louis, former home of the Bowling Hall of Fame. This paper on medRxiv caught my eye, and I immediately thought, I wonder how Brienne's campus might institute this, just as a thought experiment. I went to a small Midwestern college, go Illinois Wesleyan Titans, of about 1,900 students, and think it would be possible but challenging to implement here. UW-Madison, forget it. So what do we got here? This is COVID-19 screening strategies that permit the safe reopening of college campuses by people at the Yale School of Public Health, New Haven.
There you go.
Mass General and Harvard Medical School. Frequent screening and uncompromising, continuous attention to basic prevention and behavioral interventions to reduce the baseline severity of transmission. Daily to weekly testing with high sensitivity, specificity, and low cost, $10 to $50 a case. I mean, you know what? Undoable at Madison? No, it's not undoable. If this were World War II, you would figure out a way to do it. And this is war. This is World War C-19. So I don't accept excuses. I got a letter from a laboratory person who said, you have no idea what it takes to develop and execute a test, how many people, how much this and how much that. And I said, you're missing the point. You got to try. You can't say why you can't do it. This is war. Let's do it. And I said, I'm trying to motivate people, okay? And now we're getting these letters about how it can be done. All right. Anyway, thanks for all you do. Really enjoyed 636. Lots to learn there. My wife, wrestling with teaching philosophy on the floor, asked me the other night if I was going to TWiV it. TWiV it, I asked. Yes, isn't that what you do when you go out on the deck with your headphones in the evening? Listen to TWiV it? The podcast is called TWiV, This Week in Virology, and yes, that's what I'm doing. Oh, she said, what's a TWiV it then? I don't know, but I think it's something Elmer Fudd places hot casserole dishes on so he doesn't scorch his countertops. That is just great, isn't it?
Excellent. I approve of this joke.
TWiV it. Dixon does
actually, going back to your thing about getting together testing, I'm reminded of the Yoda quote: "Do or do not. There is no try."
Yes, exactly.
Maybe that's
right.
a show title.
Do or do not. Well, I don't want to do not.
Right.
How about just there is no try?
Okay.
And that's all. I go back to Cornell, my alma mater. I'm giving them hell every week. I'm so disappointed in you, Cornell, for saying we can't do this. We don't have this or that. Yes, you can.
They ask you for a donation so you can pay for it.
I don't have enough money to pay for it. But they do.
That.
They do. What do you think their endowment is?
Of course.
Well, and if they can't do this, then they could have school online like they did last semester.
And they lost a bundle.
Well, okay.
Oh, really? Is that the bottom line?
Yeah.
Yes,
I mean,
they did.
so your options are
nothing.
Either you
don't
do
have
it
an
right
option.
in person or you do it online. And if they don't like the do it online option, then they need to do it right in person.
There's no option.
So Columbia will be open in the fall, and the teachers have the option of being in person or online or a mixture of both. It's up to the professor.
Do the students have the option?
That's a good question. Well, if the professor says I'm doing it on Zoom, the students, but if the students don't want to be in class, well, I guess they don't come, I suppose. I don't know. It's a good question. Dixon, you're next.
Yep. Barbara writes, "Dear Vincent and TWiV team, I thought you might like to meet Kid COVID." She lists the website for this one. Kid COVID Fights Back is written and illustrated by Guelph, Ontario, Canada artist Barbara Salzberg Matthews. It is a wonderful free resource for kids and parents to better understand and protect themselves from SARS-CoV-2. It has been distributed around the world and added to the New York City Department of Education's online library of free COVID-19 resources for children. For a quick video overview of the book, check out the website that's listed here. It may be of interest to you and your listeners. My best, Barbara from River Road Creative.
It's a free book. It's not
Yeah.
free illustrated book. It's kind of like Paul Stays Home, which is from Mexico. Same thing. I wonder how we could get Paul Stays Home on the New York City Department of Education's online library of free COVID-19 resources. Anyone know? Let us know. The authors in Mexico would like to know. That's pretty cool, though. The more, the better. And I'm glad they're free. I'm glad you're not trying to capitalize.
Here.
Because I know Dixon does all the time, right?
Absolutely.
Rich. I'm kidding. Rich.
I have such sumptuous surroundings, can't you tell?
A lot of paintbrushes. Those paintbrushes are $50 a pop. I know that.
Well, they used to be. They're much more now.
Edward writes, "Hi, Twivers. Given all the bad decisions made by sports organizations that have justifiably brought out Vincent's inner grump, I thought I'd share a nice ray of sanity from the roller derby community."
Roller derby, of course.
It's got a link here. The Women's Flat Track Derby Association released its return to play guidelines a little over a month ago, which features a 7-tier ladder, which includes guidelines not only when to advance, but when to back off based on clear health metrics. We even got a nice little write-up in Wired. Gives a link to that. I saw that at one point about how the roller derby was doing it right. I've been enjoying TWiV and the occasional TWiV since April, and found the Singapore discussion on dengue to be particularly fascinating. Thanks for all you do, Edward Bitshift Adams. Just, he says, a roller derby referee. I love it.
And wrestling coach.
I love it.
It's pretty cool.
A roller derby referee. I love it.
Oh, has anybody looked at their websites to see what is their plan?
Yeah, so unfortunately, they do have the 7-tier.
Is there no touching?
But
Is that
well, there's no spectators until tier 7. All right, and the participants have a lot of things to go through, but there's not a lot of testing involved here. Unfortunately,
the roller derby premise that doesn't change. Is that right?
What do you mean?
There's elbowing and kicking and screaming.
No, it's the same thing. It's the same thing. It's just that, they have some other
So all the players have to be tested.
But they're not. There's no testing here.
Well, that's silly.
Look, what it says here is until there are widespread testing of cases and or antibodies, we do not recommend your league welcome in-person audiences. But what about the people?
What about
There's nothing
players?
about the players and testing.
Exactly.
I mean, everything else is very comprehensive, but I would say all the players have to be tested initially and then multiple times a week.
You got it right.
Got it right. I guess they're going to broadcast this, right? But anyway, it's good to see you have people. As Viviana said, you need to have plans.
They do have, okay, so I'm at page 7 of this document. They do have some, they're not specifying testing or how it needs to be done.
Yeah.
They have set these parameters. Your local government has to have removed stay-at-home orders. They have to allow public congregations of at least 50 people. They have to not prevent getting together for non-contact activities. You have to have functioning public transit, and you have to have downward trends in hospital admissions, percent of positive tests, COVID-19 related deaths, and so they're relying on competent local management, and they've got a whole thing on how to calculate maximum number of active new cases for a large city. So they're basically relying on on official testing to cover this sort of thing, which means
I don't think that's a good plan. I think
I mean it kind of punts it.
Yeah, they should be testing their players for sure.
Absolutely,
that's a lot to ask for a. I mean, this is not basketball. This is
but it's
that I don't think makes a lot of money.
Yeah, but it's not about money. It's about keeping people safe, and maybe some
ideally,
of the players have older people at home and they could infect them, right? So,
but these guidelines look pretty strict. Like I could imagine if this is a local roller derby team, I could imagine them looking through this and saying, well, if that's what we got to do to reopen, then see you in 2021.
Forget about it.
Yeah.
But do you remember the letter from Japan where their adult industry, they test them multiple times a week?
Well, that's a well-funded industry. So
it's usually one-on-one also, not multiples.
Works of pornography are often criticized, but seldom dusty. They don't have a sales problem.
Boy, it would be nice to have some sort of point of care test for an active infection, wouldn't it? I mean,
as Viviana said, it's happening. But this is what kills me. How much money are we spending on recovery? Trillions, right? Why can't some of that trickle down into testing for people?
Well, some of it is. There is a lot of new funding for these sorts of things, and it also takes time to develop.
Alan, you're next.
Okay, we're up to Andres.
Yes.
Andres writes, "Dear TWiV team, I've been. I'll just hit the highlights here with our 135 pages of questions. Vincent, your lectures have been excellent. They have served as a great review, and I'm learning a lot of new things. Would you consider releasing your exams eventually?"
Sure, no worries.
Yeah, so Andres is a PhD student.
He says just a PhD student.
Yes, just a PhD student.
Now we have a just arc, right?
Right.
It's great.
Second, it seems to be a relationship between the insufficiency of vitamin D and severity of COVID-19 or other viral-associated lung disease. Gives a couple of references. While this is not a one-variable effect, causation has not been proved and other factors remain important. Some studies point in the direction of being another important cofactor in the severity of the disease. What's your general opinion about this, and should authorities put a bit more emphasis on supplementation as a preventive of severe outcomes? So the vitamin D literature is like a lot of stuff in nutrition. The studies are hard to do, and the conclusions are often kind of all over the map. There's some evidence that this might play a role. There's not a lot of evidence that taking a vitamin D supplement pill is necessarily the way to fix it, is my understanding. And my hesitation with putting emphasis on this in public messaging might be that people would engage in risk compensation. I took my vitamin D today, so I'm at lower risk. I think this is an interesting thing to look into. I think it's certainly something that I hope somebody will be able to follow up on in studies of this disease. I don't see it being a major part of the response, though. But that's just my take.
I agree.
Yeah, I asked Daniel about it, and he said people can take vitamin D, but don't take too much because it is a fat-soluble vitamin, so you can overdose.
The intriguing thing about vitamin D is it's got its own spokes in a group called the Vitamin D Foundation or something like that, and almost all the information comes out from them, and that's really
there's an industry built around selling you vitamin D. And a lot of literature comes from people sponsored by them, and so that's part of what has gotten into.
And it's not a vitamin, actually, because you can make this in your skin yourself. So that's not a strict vitamin.
You go out in the Arizona sun, right?
Right.
Or even go trout fishing sometime when the rivers are not flowing over their banks.
So you're saying me staying in my basement all day is not good?
Not a good thing, Vincent. No. That's not a good thing.
I actually have to go out and run an errand, which will involve going into the sun as well. So
thank you, Alan Dove, turbidblack.com, Alan Dove on Twitter. Good to see you again. See you next week.
Thank you. It's always a pleasure.
See you, Alan.
Have a good week.
Now we are down to 3 of us.
You are.
All right.
And then there were 3.
Trudy writes, Dear Twivers, in follow-up to your discussion regarding the Spanish study that led journalists to conclude that herd immunity is unachievable, I have the following comments. First of all, the authors admit that cellular immunity, which was not evaluated here, might also play a role in protecting against infection. There are numerous publications, and there have been numerous discussions on TWiV about the fact that T cells may be just as important, if not more important, than antibodies in protecting against a second infection with this virus. These papers lead to the conclusion that epidemiological data relying only on antibodies may lead to substantial underestimation of prior exposure or immunity to the virus. I would also like to add that although the pandemic was bad in Spain, the mandate for quarantine was put in place in early March and remained as such for quite a while, thus preventing further spread of the virus. So like Rich, I would not be surprised if the overall prevalence in Spain is lower than in other places. There are also a couple of studies done in Italy that show that up to 74% of infected people can be asymptomatic, with about 45% of those being truly asymptomatic, not just pre-symptomatic. There are many data. Notice she properly uses the word
that's great.
Data. And somebody yelled at me last time for saying, nobody learns Latin anymore. What are you worried about? I take it all just like water off the duck's back.
I don't think they were yelling at you.
No. Okay.
I think they were
chiding me.
Yeah.
It's fine.
I don't think they were even doing that.
They were being pedantic.
If you haven't noticed, sometimes they exaggerate. Have you noticed?
Really?
There are many data indicating that people with asymptomatic infection produce low levels or no antibodies at all. They could still produce T cells. This has also been discussed on TWiV, although Viviana says she doesn't see anybody infected who doesn't make antibodies.
I thought Viviana's discussion was really great because it's. I get the impression from her that in I don't know how many, but a typical scenario. This is like a regular respiratory infection. You get infected, you mount an immune response that includes antibodies, you recover from the disease, the antibodies persist for a significant period of time. Okay? And we spent a lot of time talking about what could go wrong, and a lot of times we're digging up, rarer situations that make us worry. But I think it could be that in the vast majority of the cases, this behaves like any other respiratory infection.
In fact,
sure, but if you don't have the right test, or if it's not working well, then you're going to get some spurious results. And I think that's what she also said, that the test that she's got in her hands is the gold standard that everybody should be using. And if they're not, then you're going to get different results.
And they're giving it away, folks. You don't need to buy anything from them. You just get it. And so Trudy says, based on everything we know about coronaviruses, there's no reason for us to believe that a first infection with SARS-CoV-2 won't confer at least some level of immunity against a second infection, and that a second infection will be much milder if it should occur. I totally agree. I personally think that the fears about a second infection being more severe than the first are unsubstantiated at this point. There's no evidence for that. While this phenomenon is known for dengue virus infections, to my knowledge, there's no evidence that this exists with coronaviruses, in particular with a second infection with the same serotype, but we have plenty of evidence that a first infection confers at least some level of immunity. But however, human coronas, but cat coronavirus, there is antibody-dependent enhancement, okay? So it exists in the larger coronavirus world. That's part of the concern. On that topic, there are at least a couple of studies that show that rhesus macaques that were infected with SARS-CoV-2 were fully protected from a second infection with the same virus, and that this protection was mediated by both antibodies and T cells. This has also been discussed on TWiV. Admittedly, monkeys are not people, but these results align with what we generally know and understand about immunology and viral infection. Thank you for ramping up TWiV. I don't think I've ever learned so much about one virus in such a short time, and neither have we.
Yeah, really.
We're learning.
I would agree with her that monkeys are not people, but I think the corollary is not true.
Yes.
There's a lot of people out there.
That's a joke, right?
I was an attempt at a joke.
Dixon, you're next.
Wait a minute. Was that Trudy?
Trudy, our buddy Trudy, yeah.
Okay.
Yeah. Okay, Victoria writes,
Trudy Ray.
Hi there. I'm a student at Caltech's Bi 115 class, where we discuss viruses and their application to biological systems. I was recommended to check out your podcast, which I do not regret, and I have a question for you. In 614, you mentioned that Kawasaki-like disease has been popping up, might be driven by coronaviruses in general and not just SARS-CoV-2. I came across this paper from 15 years ago suggesting that New Haven coronavirus was associated with Kawasaki disease. She gives a reference. And it seems very possible to me that there is something about coronaviruses that leads to Kawasaki-like disease. I know that Kawasaki is a pretty new presentation, but I would be very interested in hearing any thoughts or theories that you might have regarding why there might be an association between coronaviruses and Kawasaki-like disease. Thanks, Victoria. I wish Peter Hotez was here.
Why would you want to have Peter? We can get
Peter. Peter Hotez knows a lot about Kawasaki disease.
Oh, really?
He was one of the first to even say it. What's happening now resembles, and then he went on to talk about that.
Oh, well, we can get him on.
So I think he would love to talk about this. He's got a lot of information.
Yeah,
there.
I will do that. But it has been, people have been trying for many years to sort out if there's an infectious agent associated with Kawasaki's disease. Many careers have been not gone well because of that. So we don't know. And this is an interesting one where there's, the problem is this study, they look at, I got to wait for it to load again because I had closed it. This is done in New Haven, and they have respiratory secretions from children, 8 of 11 with Kawasaki, and 1 of 22 from controls were positive for this New Haven coronavirus. They suggest that it's associated with it. It's an association, and that's as far as it's gotten. But we can let Peter talk about it.
Ironically enough, Peter Hotez spent many years at New Haven.
Is that right?
He got his degree there. Yeah,
his MD
he was
degree?
A professor at Yale, I should say. He didn't get his degree there. Take that
So
back.
correct me if I'm wrong, but my sense of Kawasaki disease is that it's essentially an inflammatory disease.
Yeah, that's right. A cytokine storm type of.
Yeah, and so given the sort of apparent most of the problems, it seems to me, serious problems with this virus result from an out of whack immune response, and so it strikes me that that's completely consistent with some sort of more narrow phenotype inflammatory disease that looks like Kawasaki disease. I mean, I'm being very general here, but there's a common thread here, which is an immune response that's gone haywire.
But not every virus infection does this. So it's coronavirus specific in a very few patients.
Well, I don't think it's corona specific, but
well, that's the implication here.
But I don't think it's corona specific. No, we don't know. That's the problem. Dixon, can you take this next very long one?
Like
you don't want to do it? You're bowing
sure,
out?
No, I'll do it. Don't do it. I'll read the yellow part, only the highlighted part. Okay.
Sure.
Julia writes, Dear Twivers, Elaborating on David's letter from episode 613 about the predictive values of tests, this is a critical consideration for serology assays in light of the immunity passport discussion, clearly everyone's favorite topic, particularly Alan's. Haha. When disease prevalence is low, even tests that look really good on paper have a remarkably poor positive predictive value. If you have a serology test that's 100% sensitive, it will correctly identify all people who do have antibodies and do not miss one single, or it has no false negatives. If that same test is 98% specific, it will correctly identify people who do not have antibodies 98% of the time. Only 2% of the time will the result tell if a person, let's try this again. Only 2% of the time will the result tell a person who doesn't have antibodies that they do, or a false positive. This sounds pretty good, all in all, right? Here's where the disease prevalence throws at the wrench in. Say you test a population of 1,000 people, but only 2% of them have actually been sick. The test will correctly identify all the true positives, 20 people out of 1,000. It will also correctly identify 98% of the true negatives, 960 people, but it will give 20 people a false positive result. Isolating just the people who got a positive result reveals the problem. We have 20 people who got a positive result and were truly positive, and 20 people who got a positive result but who were actually negative. Who was who? The positive predictive value is true positives divided by all positives, 50%. A test that's right only 50% of the time is basically useless on an individual level. Most of us, outside of maybe New York and a few other places, are probably in a pretty low prevalence area. Spain, one of the hardest hit countries in Europe, just conducted the first phase of a major serology study involving 60,000 randomly selected persons. The average prevalence was 5%, 95% CL, 4.7% to 5.4%. Now, that's not a very large standard error for the amount of testing that they did, but it's a lot of people, right?
Yeah, they did 60,000 people.
60,000, so 5% of that is a big number.
It's a study we talked about last time, right?
Yeah.
Right. The upshot for me, we have a long way to go before any of these results mean anything about who can consider themselves safe for a future infection or from being a vector of transmission. Keep up the great work, please, Julia. P.S. Should I read that part?
No, you have to read it. No,
okay.
It's okay. The thing is that, 98% isn't good enough, right?
Right.
No, you're absolutely
especially in low seroprevalence areas. Yeah, and
it would be better off if you had a lower specificity but 100% sensitivity.
Right.
In a broad sense, applied broadly, it can give you an approximation of the seroprevalence. But as he's pointing out, on a case-by-case basis, it's almost useless because you can't really rely on it.
Exactly right.
There are things that you can get from serology studies. First of all, you want to know if reinfections occur, so that helps to tell. Although, if 50% of the people weren't actually infected to begin with, it's not going to help a lot. But for me, it's just to know how many people in a population have been infected in a particular area.
You can afford the error, but I worked on a test for about 3 years for identifying trichinella in pigs. And they were desperate for a test to be 100% accurate because the cost of a pig was pretty high if you missed the diagnosis. So it eventually didn't succeed because of some other reason.
I guess the key here is you can't use serology to say you can go back to work.
Positive or negative.
You can go back to work or not. That doesn't work.
Yeah, you can't.
You can do it for other epidemiological reasons, but you can't give immunity passports. I think that's the bottom line, and that's what Viviana said. That's it. No matter how good your assay is. Rich?
Eric writes, Dear TWiV team, I watched all the 2020 lectures from Vincent's virology course. It was fascinating. Here's what I learned. Uh-oh. Here's your test,
No, you
Vincent.
shouldn't say that. Vincent should say that.
1, the COVID-19 virus, or the virus that causes COVID-19, is an RNA virus, so on average mutates 1 or 2 bases on every reproduction. 2, when the virus kills its host, it also dies. since a dead host does not cough or breathe.
So did you teach them that too?
3, Darwinian selection will then result in a less lethal virus over time, or the virus will not survive. 4, the July COVID virus is not the same as the February or March virus.
I didn't teach him any of this, only the first point.
You're in denial.
Yeah, we can go back over these and refine the understanding. So why is this simple virology fact not discussed more? Today we hear about cases rather than deaths. Could it be that those in power and in the media have an agenda to keep the level of fear high for reasons other than spreading the truth?
The people in power don't even understand any of this, so that's
true.
And there is certainly a certain element of bad news sensationalism selling news. Okay, that gets mixed in with the science, and that's frustrating. That was our discussion of the serology result last time. There's a lot of inflammatory news reporting, but in the case of the news media, or in the case of the news media that I personally respect, the motivations really are to sell news, not necessarily to influence the population with some other agenda. At any rate, when the virus kills the host, do we really want to do this? When the virus kills the host, it also dies while it was never alive. Okay?
Right.
But
the point here is that transmission has already occurred before the host dies, right?
Yeah.
So you can't say that it's going to be selected for less. I mean, what I do discuss in my course is that over time, viruses may become less virulent because the longer a host lives, the more likely it is to transmit. All right. But that's going to take a long time, not 3 or 4 months.
No, we're talking about years, generations of people, generations of hosts and viruses.
Now, Rich,
I think
in defense of Eric, I do teach them the Australian experiment, which in 1 year, the virus changed. So, but that's rabbits who are multiplying a lot faster than people.
Actually, I'm not sure that the attenuation of myxoma happened as rapidly as a year. It did attenuate over time.
But the first year, there was just a few rabbits who didn't die, and they survived.
That's right.
That was it.
That's right. That's right. I think, my conception is that it is generally true that when you have a spillover, you have what I think of as a non-equilibrium situation. Where if circumstances are a certain way, the virus can be transmissible and quite often very lethal because it hasn't equilibrated with the host. And over a long period of time, there's a tendency towards equilibration. This is great. My grandson, 9 years old. I was talking to him today. We were talking about this stuff. And he said, let me see, we're talking about events that occur with chronically resident viruses on rare occasions. And he said, "You mean like PML?"
Wow.
And I'm going, whoa.
Where did you get that from?
Right? Me. Okay. And I said, "Do you remember?" Because I tried to get him to be able to say repeatedly, progressive multifocal leukoencephalopathy. Okay. And I got to work on him a little more so he can spill that one out. But he had the whole thing. People wanted to be reminded which virus was that. I said polyomavirus. And Porter says, "Yeah, that hangs out in your kidneys, right? And doesn't do you much harm." Yeah, Porter, that's right.
Very good. Very good. You're doing a good job there, Dr. Condit.
Well, it's hard to, not much gets by Porter. I noticed some time ago that he asked a lot of questions and never asked the same one twice.
Nice. We met Porter in TWiV 500.
Yeah, he's been on the show a couple of times. He's signed in with us a couple of times.
All right. Brian writes. So Brian writes, "You rock." You know what that means, Dixon?
That's a university, isn't it?
Recently, I heard your team express doubts about aerosol transmission of SARS-CoV-2. Like the WHO, they pointed out large droplets comprise part of the total volume of expelled respiratory droplets in sizes greater than 20 microns. virus presumably in the larger ones. But evaporation time for droplets, any of these sizes, is very short because they have an enormous surface area compared with their mass. For example, a 12 micron droplet will take approximately 0.02 seconds to evaporate. A 25 micron droplet will travel perhaps 5 centimeters before it shrinks to 5. Wow. Humidity, temperature, mucin content of droplets will all have an effect on evaporation rates, but virus on a larger droplet will soon enough be on a droplet nuclei, right, as small enough for air resistance to take over. So I assume you're right. Virus are in larger droplets, but those aerosols are dynamic and become flyers. Some references below. So I totally agree. This is absolutely right. The larger droplets will evaporate, and they could potentially go farther, and that may happen. That probably happens in some cases, as long as the virus is not inactivated in the droplet.
And I think there's accumulating evidence that some sort of aerosol transmission does happen over longer distances than you would expect for large droplets.
Yeah, this has been around the news this week, WHO. We'll get to that next week sometime. P.S. Compartmentalized epidemiological models don't include complex households where most transmission occurs. Here's the thing. It's probable that within household behavior is a more significant transmission variable than outside household. Absolutely.
Absolutely. I think that reminds me of the letter we had from the MedTech in Israel, who pointed out that the social interactions in households in Israel's are quite close. Okay.
Yes, that's right. Dixon, you are next.
Juan writes, Dear TWiV, it's 33 degrees C here in Milan, Italy, around 91 F, I believe. We passed the peak of COVID a few months ago, although the number of daily cases are still near the 100s. Many of these, however, are only found after serological test is done to seemingly healthy individuals, which after finding they have antibodies are tested and turn out to be PCR positive. Regarding these weakly positive individuals, as the authorities here are calling them, the head of one of the most important hospitals in Milan has claimed that the virus is now less powerful in English. He gives a reference here on the internet. Claims look intentionally controversial. I don't give them much importance, but the following related question has come to mind. I've heard several times that the threshold, or whether there is one, for infection with SARS-CoV-2, and indeed most viruses, is not known. For the sake of the question, let's assume such a perfect threshold exists. To me, that means that below the threshold, we get virus particles in our bodies, but do not get infected. How would these particles be cleared in a naive individual? Does the body learn to eliminate them? Can a person gain a certain degree of immunity of any kind by being exposed to very small quantities of an infectious virus? Can this be the way that COVID becomes another common cold? I guess this is impossible to study in humans rights and all of that, but I wonder whether the answer to these questions are known, maybe for other viruses in animal models. I'm just a particle physicist, a small particle or large particle physicist, and my knowledge of anything bigger than an atom is very limited. Now, thanks to you, I'm learning to appreciate how stupidly exciting these non-alive creatures are. The non-COVID episodes are discovering me so many ways, are revealing to me so many new ways I can die, but also many exciting new things that I can't stop listening. Thanks. Note, I guess you guys already have access to this kind of data, but I wanted to point out the Italian Epicentro website, which collects the official data since the beginning of the outbreak. Oh, that's very interesting. Thank you so much for all your work. Juan.
Yeah, this was pointed out months ago by Valeria Cagno when I was talking to her. So a couple of things here, Dixon and Rich. Weekly positive is BS. This was a month ago. Daniel dealt with this. You know, these people are at the end of their infections and there's very little virus in them. So this guy thinks it's weekly positive. It's just wrong. So this idea that you can get a small amount of virus and you get immunity, this is not right. So small amounts of virus are probably dealt with by innate systems and including interferons and NK cells and so forth, and there's no adaptive response. So, you know, if it were that simple, we would give a vaccine of 1 PFU and it wouldn't kill anyone. It would give immunity. But that's not what happens. No. We have to change the virus in some way to make a vaccine. So I think that's the bottom line there. Yep. Okay.
Mm-hmm.
Rich.
John writes, hello, TWiV crew. I'm a geriatric doctor. I believe that's a doctor of geriatric people.
Yes. Old people.
With a long-standing...
Or he's an old doctor.
with a long-standing running/biking habit. During those hours of Zen, it occurred to me that the risks of SARS-CoV-2 transmission reception are mostly related to breathing. I've been holding my breath while passing others on the trail. One can stop breathing for about 20 feet on approach and resume 20 feet in passing. In my simplistic model, this assures adequate protection. Now, this will not mitigate the ill feelings of others, and I have no objective evidence, but this helps me feel better. Thanks for all you do. John's, of course, an MD.
He didn't say he was wearing a mask or not, but he presumes he's not, right?
I think if it makes you feel better, go for it.
This is a good point.
It's not going to do you any harm.
If it feels better. You want to hold your breath. It seems to me if you're biking hard, that's tough to do, but...
Yeah. Well, my wife and I still, actually, even more than ever, walk. We're turning into walking maniacs. And I've noticed that around our extended neighborhood, it's not a real dense population of walkers. People are very respectful. People aren't wearing masks because they're outside, but people move to the other side of the street. Yeah, we just stay out of each other's way, clear out of their way, way over the 6 feet.
All right, let's do one more round, gentlemen.
Okay.
Luis writes, Dear Vincent, Rich, and Kathy, I'm a recent but now avid listener of TWiV. Your podcast is an island of wisdom, reliable information, and even entertainment in a time of mounting uncertainty, ignorance, and chaos. I want to call your attention to the comment that was published in Lancet along with the original paper on the Spanish SARS-CoV-2 seroprevalence. If you had seen it, you would have read, quote, in light of these findings, any proposed approach to achieve herd immunity through natural infection is not only highly unethical, but also unachievable, end quote. So the press was right on this one, and I was disappointed that you were so dismissive when you did not see the accompanying document. I don't give a damn about I don't care about a commentary, okay?
Right.
I don't read them. I don't care what someone else's opinion a science writer is necessarily. So I don't read them. I read the paper because I can understand the paper. And I don't agree that it's unachievable. I mean, the headline basically said that herd immunity is wrong, okay?
Yeah, I went back and I read that in a little more detail to see where that came from. And yes, that word unachievable came from a commentary. And I think there was some commentary from scientists who they interviewed who were putting together the notion that there was, after this wave in Spain, a low level of seropositivity with the, I would say, idea that immunity can wane, and saying that given the low seropositivity and the waning immunity, you put those two together and herd immunity is going to be unachievable. I don't agree. The data don't say that. Okay.
I mean, you even searched the manuscript. I mean, that's what I care about.
The manuscript certainly.
So I think it's inappropriate for the press to pick up something from a commentary instead of the actual paper itself. That's
and make that the headline.
Yes, because that's someone's opinion. All right, on the paper, not the author's.
What do you say, Dixon? Trust the science, not the scientists.
You got it.
So look at Corona Queens. They have 68% immunity.
Yeah.
Maybe they're close. Now, they had a lot of infections there. It wasn't nice. So this statement that is unethical but unachievable is simply not correct. I think it depends where you are. And we're not purposely doing it. We don't have any choice in many cases, okay?
Right.
Also, he points out, which is a good point, the fact that someone was infected and recovered does not at this point guarantee that the same person cannot be infected within months. Well, we've mentioned this now over and over again, but we're assuming that this is acting like most viruses, but who knows?
That whether or not you can get reinfected and to what extent that happens and whether or not you get serious disease, those are all unknowns at this point. I was impressed with the conversation today that said that the people who were PCR positive, 99% of them made antibody, and that antibody over months was stable. Okay, so it's not like, you know, this focus on the bad outcomes leaves the impression that people are not necessarily making a strong antibody response, and that's decaying. I don't think that's the normal situation. We don't know a lot of this stuff, but I don't think that we can say that that's the normal situation.
So he says, to be clear, I'm with the authors of the comment on this, as I think considering natural herd immunity can be achieved not only is ill-informed, but highly unethical and has real-life consequences. But that's what happens for many other viruses, you know, and especially before vaccines. And I don't know why it's unethical. It's not like we're trying to do it. In addition, I'm surprised by the argument that perhaps seroprevalence grossly underestimates true exposure to the virus. While I am convinced that cellular immunity is perhaps more important in this infection, the ones where cellular immunity controls replication quickly, not only to do better, but develop lower antibody titers, I do not see that T-cell responses occur without also mounting an antibody response. In fact, looking at T-cell responses, CD4s are the ones that are invariably present on the exposed patients. These are the type of T-cell responses that correlate with an antibody response. On the other hand, a recent paper in Cell shows that among unexposed individuals, 40 to 60% show SARS-CoV-2 reactive CD4 positive T cells, which suggests cross-reactivity with common cold corona, but which significance is unclear.
Right.
So let me go back to this. I am surprised by the argument that perhaps seroprevalence grossly underestimates true exposure. I don't think it grossly underestimates it, right? But
no, and I don't think we said that. I think we said, so first of all, once again, I was impressed today with the observation that the vast majority, if not all of the individuals at Sinai who were PCR positive were antibody positive.
Yeah.
Because we have done a couple of papers where, now this was a situation where they were looking at asymptomatic individuals. Okay. But we've seen a couple of situations there where there was a serious fraction, a significant fraction of asymptomatic individuals who mounted a T cell response, but not an antibody response. But that's a very select population.
Yeah.
So, I mean, given that is possible, then it is possible there may be some of those in the population, and you would therefore underestimate the true infection rate by just measuring seroprevalence. But I don't think we said it was grossly underestimated. As a matter of fact, I think said if, a maximum of maybe twofold, but it's probably not more than 10 or 20%, if at all.
Dixon.
Yes. John writes, Dear Twivers, I thought you had more to read on that. I wholeheartedly agree with Professor Racaniello's outburst about the CDC trying to kill us all with their recommendations against COVID entry testing of returning IHE students, faculty, and staff. Maybe it is time to hold a listener contest for what the acronym CDC stands for. My entry will be Centers for Disease Contagiousness.
Did I have an outburst? Do I do that?
Let me think. I refuse to answer that on the grounds that it might incriminate me. No, I think you tend to rant sometimes. You might have ranted a little bit about the dismantling of the integrity of the CDC by appointments from certain political leaders.
I'm just joking. I do. I have strong opinions, and
no, you're allowed opinions.
All right, one more.
We don't have to agree with them.
Let's do one more, Rich. No, you don't have to. Of course not.
Would this be Eli?
Yeah.
Eli writes, Dear Doctors TWiV, I'm really glad I discovered your podcast a few months ago and hope to continue listening long after the current crisis is a historical footnote. Bravo. I appreciate that your explanations are usually clear to non-scientists, and especially that your expertise is combined with an admirable dose of we don't know. I wish more people in public understood that an honest admission of ignorance builds credibility. Good. I hope you don't mind practical questions about how best to proceed in business. My spouse and I run a used bookstore and coffee house in the northwest corner of Washington State, 67 Fahrenheit and sunny. We only occasionally get above 80. And at 85, the town mostly shuts down. Thankfully, air conditioning isn't really a big thing in these parts. After a few months of being closed to the public, we opened our doors again last month. Our biggest issue is how to buy used books from the public again. Our shelves are lighter than we like, and there is an embarrassing number of holes in our stock. We're also fielding no less than a dozen calls a day from folks who want to sell us books. Our current plan, once we find available space near the shop, is to quarantine the books for 72 hours before processing them and another 72 before shelving them. That would be a total of like 10 days, right? Unfortunately, securing a space is proving difficult, and we're questioning whether those precautions are wise or necessary. And even if we have the space, that seemingly simple 6-day rotation is a logistical nightmare. We have great ventilation. We renovated the building a few years ago and invested in an air handling system that can exchange, not recirculate, the air in the shop every 90 to 120 minutes. It has been set high since March. When customers are present, the doors are open and the bathroom fan is always on. Masks are required of employees and customers, and we insist that everyone don fresh gloves or use hand sanitizer as they enter the space. Bottles of hand sanitizer are scattered throughout the stores. Commonly touched surfaces are wiped down with alcohol or quaternary ammonium solution several times an hour. While plexiglass is awfully dear these days, our workaround has drawn many complaints. See attached photos.
Compliments. You said complaints, not
I'm sorry.
compliment.
Compliment. I'm sorry.
Oh, he's got a nice plexi in front of the cashier. Nice store. I got links to the bottom to the pictures. Very nice. Okay. I'd like to visit and have a cup.
We listened. We limit customers in the store to about 25% of normal capacity. All coffee and food is for takeout only. We could legally allow a very limited amount inside of seating, but decided that unless a customer can drink a latte through their mask, they must take it outside. Blessedly, no one has tried. Our staff is taking safety as seriously as we are and understand that anyone who feels ill at all, they're not to come to work. And will be paid for their shift anyway. We also check every employee's temperature before they can begin their shift. Until this week, we quarantined and disinfected all cash. My wife was going nuts with the bags of bills. We noticed that we're the only place in town doing it. Still, anyone touching money is wearing gloves, and no one who prepares food or drinks touches money. That's the good stuff. But there's other stuff, largely out of control, that worries us. Our small staff ranges in age from 21 to 75. We're confident that none of them are engaging in overtly irresponsible behavior, but several are young, living with roommates, and falling in love. Good for them. We have to discreetly inquire about parts of our employees' lives that should be none of our business. To our knowledge, none of our staff intimates have fallen ill, but there have been cases of COVID in the same social circles they swim in. We fear it's just a matter of time before someone on the staff tests positive. We don't have the same issues with close quarters that a lot of retailers and restaurants do, but 6 feet just isn't possible much of the time. We're sanitizing computers between users, and only my wife and I answer the phone, but there's no practical way to never miss a spot and never be only a couple of feet apart. Maybe because we're a bookstore, or maybe because we live in Lake Wobegon, our customers skew heavily to the right of the bell curve. We've been remarkably free from knuckleheads. But the nature of a bookstore means that people are going to browse, pausing in front of shelves, touching multiple books, generating an invisible cloud of whatever their bodies are shedding, despite the mask. Even if a mask blocks 90% of virus, still means that 10 minutes in one spot is going to leave some cloud that someone would leave after 1 minute without a face covering, right? Right. There's always going to be some virus around. We're proud of our governor's response, but shutting down once nearly broke us. If we close again because the state orders it or because someone on the staff gets sick, we might not be able to open again. To say the least, I am unimpressed with folks who are blasé about masks and other precautions. So back to the original question. How do we buy used books? Our operating assumption is that every book someone tries to sell us was sneezed on by someone with a full-blown case of COVID 10 minutes earlier. That's a little extreme, but okay. Of course, that isn't true, but it's a reasonable assumption to build protocols. Is it a reasonable assumption to build protocols on? Besides quarantining them, we've toyed with bringing out the ozone box we sometimes use to deodorize stinky books. But using that comes with its own set of safety and logistical issues. It seems surface transmission is rare, but not impossible. Paper doesn't seem to hold live virus very long, and most book covers these days have a thin layer of plastic. We've always wiped down many books with sanitizing wipes anyway. Extending the habit to every book is doable, but expensive at scale. On the other hand, I feel like we're already doing so much that quarantining books is like installing seatbelts on a train. Good analogy. On the other hand, because of the nature of our business, the inherent risks of having employees who live in the world, it seems like anything beyond the basics is like putting mosquito screens in the windows of a house with no roof. Oh. I'm really bad at quantifying risk. What can we do that's really helpful, and what is not theater? What are we missing? What would you do? Thanks, y'all, for all. Thanks for all y'all do. Wow!
Screen door on a submarine.
I think. What a beautiful store.
It's really nice, isn't it?
Yeah, I'm looking at the picture. Let me look at this other picture.
It'd be a shame if it's shut down. I love bookstores.
What a beautiful store.
And I love the wood. Yeah, it's really nice. Black ceiling and walls.
You know, I think you're doing everything you can.
I don't think you should worry about the books, frankly.
No. I mean,
I think it's a low risk. There's very little transmission by touching stuff. It's mostly people in a crowded area, right? So I think forget the books. Just put them on the shelves. Have everybody wear face masks. You're doing that. And you're not going to be able to keep them apart. Don't worry about it. I think people will keep apart from other people as they see people in the aisle to move away. They'd naturally do that, and I think that's all you have to do.
I think people are going to go out and go shopping and that kind of stuff. Have to accept some personal risk for what they're doing. Okay?
I would say put up a sign that says the books could be contaminated at your risk. Wash your hands when you get home.
Right. And make your expectations of physical distancing and mask wearing and stuff clear, and insist on some adherence. It sounds like you have a cooperative clientele. You're doing the best you can. They have to accept some risk as well, and we got to get on with our lives.
And I think when fast, cheap tests come down, which they are, you should do them on your employees multiple times a week. In fact, next week, we're going to have someone on who's going to talk about developing a fast, rapid test. That should be fun.
And I think you have the most attractive plexiglass screens
They're nice,
I've
aren't
seen
they?
Anywhere. Yeah.
With the black frames around them. Yeah, someone wrote to say that plexiglass is in very short supply because everyone wants it for this kind of retail thing. All right, gentlemen, that'll do it for TWiV 638. Show notes and letters at microbe.tv/TWiV. Questions and comments, TWiV at microbe.tv. Cold hard cash, microbe.tv/contribute. Now, I'm just being silly. I know. We appreciate your donations, all of you who do. I think that's great. I really appreciate that you want to support the dissemination of good knowledge, countering the miasma of anti-think. Someone wrote in, said, if you make a T-shirt, a TWiV T-shirt with that on it, I'll buy it.
Right.
I think I'll make one.
Oh, do that.
Microbe.tv.
Maybe we should all wear it.
Tribute Dixon de Pommier is at trichinella.org and the livingriver.org. Is it .org, Dixon?
I think so.
Let's look it up, the livingriver.org. Ah, it's such a beautiful site, Dixon.
It is. It's all your
stream ecology for trout anglers.
Yep. No, you were a big help.
Thank
you, Dixon. It wouldn't be
Dixon.
nice without your help.
Thank you, Dixon.
You're welcome.
Oh, when you ask for help, I usually give it.
No, you've never refused.
I may crank a lot, but
no, not during the work process. You do not.
Rich Condit is an emeritus professor, University of Florida, Gainesville, currently in Austin, Texas. Thank you, Rich.
Fair enough. Always a good time.
I'm Vincent Racaniello. You can find me at virology.blog. I'd like to thank the American Society for Virology and the American Society for Microbiology for their support of TWiV, and Ronald Jenkes for the music. This episode of TWiV was recorded, edited, and posted by me, Vincent Racaniello. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.